Sir, this is the old CV, not the new one.
I have this one actually.
Okay. विशाल सर को शेयर कर दीजिए. So because संकेत सर और मेरा CV थोड़ा बदला हुआ था तो.
Okay.
If you want, I can run it from my side. Don't worry. If you
Yes,
want,
that
sir, I
will be
can run it.
better.
I'll run it from my side, sir.
Yes, you share your screen. I will read it.
Just let me just see it once. Sharing part. Just give me 1 second, sir. I'll share it and then you can see. Okay.
So you will be showing all the 4 CVs of the coordinators, right?
Yes. Shali ma'am has just now updated her CV. I'll add that also. I'll run all the CVs. Don't worry, sir. I'll run all the CVs throughout the program.
I will read the CVs.
No problem.
Yes.
I'll run.
So we are live in 3, 2, 1. We are live now, sir.
Good evening, everybody. Myself, Dr. Vishal Dave, Medical Advisor at Intas Pharmaceuticals, based at Head Office, Ahmedabad. It's my privilege to welcome you all for this webinar, iHera webinar on the topic Tails and Neck, Understanding Sperm Anatomy and Function. First of all, I would like to introduce our 4 coordinators for this webinar. I would like to introduce Dr. Nidhi Singh. Dr. Nidhi Singh is Medical Director at Tejasvi Women's Wellness and Maternity Center. She is Senior Consultant Embryologist at Panakia Hospital. Next, ma'am.
Just not sure. Give me a second, sir.
She's also Professor at Panakia Institute of Intern Research. Madam, your screen is not visible.
Yes, just a minute, sir. Sorry. So next.
Yes. Dr. Nidhi, Miss Yoshita Tanwar, she is currently working as a clinical embryologist at Oasis Fertility Center, Karnataka. She has also worked as Senior Embryology and Laboratory in charge, New Life India Fertility Clinic Private Limited at Delhi. Miss Nancy Sharma, she is Senior Consultant Embryologist at Department of Reproductive Medicine, Jindal IVF and Sant Memorial Nursing Home. And last but not the least, Dr. B. Ananta Lakshmi, she is Consultant and Senior Clinical Embryologist at Janni Trichy Fertility Center. She is the first woman from certified as reproductive embryologist from American College of Embryology with highest score ever in the exam. Now over to you, ma'am, Dr. Nidhi Singh.
Good evening to all. On this chilly evening, a very, very warm welcome to all. And it is the year ending, but we will be talking about the beginning of the sperm, that is the head and the tail, in a topic which is missed by all, but it's a topic of interest to all. So I would like to invite our founder and co-founder, Dr. Ved Prakash sir. Sir is a scientific director of Sperm Life Advanced Embryology Lab in Delhi. Sir does not need any introduction to this fraternity, and I welcome sir. Good evening, sir.
Thank you. Thank you.
I invite Dr. Professor Sanjay Shukla sir. Sir is a senior consultant clinical embryologist. He is the co-founder of iHera and adjunct professor at Clinical Embryology Department of Reproductive Medicine and Surgery, Mahatma Gandhi University of Medical Sciences Technology, Jaipur. Welcome, sir. Dr. Charda Joshi sir. Sir has experienced more than of 2 decades, has doctorate in life sciences, is currently the medical director of Genes India ART Bank and lab director of IVF unit at R.D. Gardi Medical College. Welcome, sir. I invite Dr. Charda Joshi, sir, to please welcome all the faculties and the members.
Yeah, very good evening to all. It's my privilege again to invite you, all of you, for this fantastic another webinar which we are regularly conducting under iHera. And as we all know, this from last few years now, the sperm has been given the due importance. And still today, it's always there is a question we always have this where there is a fertilization will happen or not, or it is because of oocyte or sperm. So we just thought of in today's webinar the topic is decided on this basis only, that let's know the in-depth of what sperm give and what is the role of sperm in fertilization, what is the abnormalities which is related with the fertilization and all. So we have experts, speakers with us, and we have expert talking on this. So once again, I welcome all of you to listen and to understand and to expand our knowledge. And meanwhile, any questions or any doubts are there, you can always free to ask in between, and that will be taken care of. So over to you, Nidhi, again for further proceedings. Thank you.
Thank you so much, sir. So I would like to invite our first speaker of the day. Is the CV visible to all?
Yes, it is visible.
Sanket sir is a senior consultant embryologist of Hegre Fertility, Hyderabad. He is SC certified clinical embryologist. MSc, he has done his MSc in genetics and MSc in clinical embryology from University of Mysore. Sir has many feathers to his cap, and he is a wonderful teacher and a very wonderful senior. I welcome you, sir.
Thank you so much, Nidhi. Yeah, let me share my screen.
Yes, sir.
Is it visible?
Yes, sir.
Yes, it is.
Well, yeah, thank you so much. Last time we spoke about the sperm with respect to cap, without a player, with cap or without cap. So now in this webinar, we're going to deal about the rest of the part of the spermatozoa, that is the neck and tail, and the dynamics of the sperm, how this neck and tail are very, very much important with respect to the fertility potential. So having said that, again, I'm going to start this lecture also with the same statement of mine, where the sperm morphology, like or an impressive period of like spanning more than 100 million years, has been constant. Like, this is like very captivating illustration of evolutionary stasis. Having said that, yeah, first, let me, before getting into the lecture, let me burst two myths with respect to spermatozoa. So there are two myths regarding the Leeuwenhoek relationship to the discovery of the spermatozoa. The first and foremost would be like, initially we all know that like he is the first person to observe the spermatozoa under the microscope. In fact, it is not right, but the first person to see them under the microscope will be Johann Ham, who was a medical graduate, a medical student at that point of time, saw this like spermatozoa under the microscope, from the microscope which was built by Leeuwenhoek. Like, initially he saw and he felt there are animalcules. They ended up with the process of putrefaction, like because he got the sample from the gonorrhea patients, he found that it is like because of putrefaction you get to see these animalcules inside the seminal fluid. So, but in fact, it is not Johann Ham, but Leeuwenhoek was the first person to consider them as a normal component of semen, and also provided the first detailed description of spermatozoa. Bursting another myth is like, it revolves around the Leeuwenhoek describing homunculus in spermatozoa, right? So it is not like Leeuwenhoek or not the person like Nicholas, like who is famously known for a drawing of homunculus. This actually revolves around a person who made a first claim, like, his name is Palantate, which was later revealed to have which is a huge practical joke because of this historical misconceptions. So after busting these two myths, let's jump into the subject, where if you look at the schematic illustration of the ultrastructure of human spermatozoa, it has principally as two regions, like a head region as well as the tail region. The tail region is again subcategorized into mid piece, principal piece, as well as terminal piece or the end piece. So if you look at the in detail location wise, organ like area wise, you get to see acrosomal cap in the beginning, then you have acrosome, Then you get to see the nucleus, the nuclear membrane which is surrounding the nucleus, and you get to see the proximal centriole and the mitochondria, mitochondrial sheet, and the dense fibers surrounding the axoneme and the annulus. Finally, you get to see the axial filament, which is just surrounded with the plasma membrane. Having said this, if you look at the human spermatozoa under the scanning electron microscope, you get to see something like this. If the magnification is around 10 micrometer, you get to see the entire length of the human spermatozoa starting from head to toe. But whereas if you magnify a little furthermore, you get to see very distinctive regions like head, mid piece, and as well as the principal piece. And if you take a very classy black image of cross sections and as well as longitudinal sections of cross sections of the spermatozoa, you get to see a very distinctive axoneme, which is having a very highly conserved 9+2 microtubule structure, and which is surrounded by a lot of mitochondria surrounding them. If you take this is like with respect to cross section of the midpiece, and if you take a longitudinal cross section, you get to see a lot of fibers, rod kind of things like which is surrounded again by the mitochondria. So it would be wrong if I don't include this point here, because during my I still remember this during my first classes with respect to developmental biology in my master's. My professor said me this in the very beginning of developmental biology classes, where Theodore as well as Bennett initially identified the centriole, right, during their studies with respect to the fertilizations in sea urchin, and also a parasitic nematode worm, that is sea urchin, somewhere between 1876 and 1900. So this was the initial step where the thing about centriole got started, has begun, and this was very much accepted that sperm centrioles have a very essential functions in mitotic as well as meiotic cell divisions, and as well in also in early sperm formation during spermatogenesis, and also in formation of sperm tail and linking the sperm tail to the head, and also establishing a very distinctive microtubule organization center of the zygote. And even early spermatids of vertebrates were found to possess two centrioles, initially identified as the proximal as well as the distal centrioles through the basic cell staining techniques by Ritter and Thomas Harrison Montgomery. And also during this spermatid maturation, the distal centriole initially attached to the cell membrane is pushed back, and it is invaginated through the nucleus. It is the distal centriole is pulled forward towards the flagellum. As the flagellum develops, it is pulled forward and it is invaginated inside, whereas the proximal centriole is positioned near the nucleus, right? So still, which is away from the membrane and located far from the distal centriole. So this distal centriole has a huge important role with respect to the flagellar formation as well as the motility. If you look at it, if you categorize broadly them into early spermatid as well as spermatozoa, and usually the early spermatid has encompassed of two by the cell membrane, which has a round nucleus, and a pair of canonical centrioles like proximal centriole and distal centriole, and a flagellum made of axoneme, the very primitive structure of flagellum, which is made of primitive structure of axoneme, and as well as the cilia membrane. And you get to see a transition zone which separates the centrioles from the flagellum. Excuse me. So the proximal centriole gives rise to the centriole adjunct, as well as in contrast, what you get to see in the developed mature spermatozoa is with a very flat, elongated nucleus rather than the round nucleus, a canonical centriole, that is proximal centriole, and an atypical centriole, that is the distal centriole, and also flagellum made of axoneme, cilia membrane, and a decreased amounts of cell membrane, right? And you very clearly, this is marked by this annulus transition zone here, which separates the flagellum into two parts, like a mid piece as well as the principal piece. And if you look at the early, if you take a cross section of it, and if you look at the early spermatid centrioles as well as the matured spermatozoan centrioles, initially in the stage of early spermatid, The centriolar complex of early spermatids contains a pair of canonical centrioles, that is proximal as well as distal, and an axoneme as pictured from the side view. But a cross-sectional view of this proximal and distal centriole displays a very it consists of 9 triplet microtubules instead of double microtubules. Like it has A, B, C microtubules, and is made up this triplet microtubules are connected by AC linkers, right? AC linkers which attach the A tubule with to an adjacent C tubule. And the upper part of this proximal central and distal central has a very scaffolding structure that connects the microtubules to the microtubules of the centriole. And on contrary, if you look at the mature spermatozoa, it is comprised of 9 doublet microtubules here, 9 doublet microtubules where you get to see like alpha A and B microtubules, and outer and inner dynein arms connecting the A tubule to one of the B tubule of an adjacent doublet. So this proximal centriole gives rise to a centriole adjunct. And coming with respect to the functions of the sperm centrioles, yeah, you get to see this in the neck region of the, mostly in the neck region of the spermatozoa. If you look at the gross anatomy of the spermatozoa, this lies either towards the nucleus and almost like before starting the actual principal, the tail region. So this is where it is located, and it has 5 distinctive functions, like starting from early, if you look at it, it is very much involved in the cell divisions during early spermatogenesis, so mitotic as well as meiotic divisions. And it is, as like I discussed in the earlier slides, it is very much involved in formation of flagellum during spermatogenesis, and it has very distinctive characteristics between early spermatid as well as mature spermatozoa. And it also has a role to connect this head to tail. This is this forms a link between the head and tail, and also it is very much involved in the motility regulation, which is a crucial part of the sperm, which enables the sperm to move forward, like progress forward. So this has a very unique role to play in the motility regulation. And also finally, post fertilization also they get to do some job, like see, usually in humans at least, the centrioles are contributed from the male rather than the female. If you look at it, mice, it is like female again. It is species dependent, but in case of humans, the centrioles for the early zygote to form the aster formation in the zygote is because of the centrioles contributed from the spermatozoa, not from the oocyte. Now, these are the 5 important functions of sperm centrioles. Having said that, yeah, this is like kind of repetition from my previous lecture, because it is again overlapping a lot of phases. If you categorize them into 16 steps, Golgi, cap, acrosome, and maturation phase, mainly the manchete as well as the axoneme starts developing at the 8th step to 16th step, and finally you get to see fully formed tail at the 16th step, which is a maturation step. And yeah, spermatogenesis unfold, like as I said, across 16 steps and characterized by changes in the nucleus and acrosome as well as elongation of axoneme during the steps 1 and 8. So this formation of the sperm tail shares a lot of molecular mechanisms with cilia, indicating a lot of common points with respect to cilia as well as flagellum. And also motile cilia and the sperm tails exhibit a very highly conserved, evolutionarily conserved microtubule structure, that is 9+2, emphasizing their structural similarity of the core. And also, as I said in the earlier slide, steps 9 to 14, this 9 to 14, involved in the creation of this manchete and also transient microtubule platform that's shaping the sperm head and aiding protein transport to the developing tail. So this protein transport occurs in 2 different steps, like they're broadly categorized into IMT as well as IFT. So IMT is like the proteins which are docked towards this intramanchete transport, which are has a specific and distinctive role to do. And similarly, the proteins which are docked towards, like categorized towards the IFT, like intraflagellal transport, have a very different role to play. So let's see in the upcoming slides. So IMT plays a very crucial role in storing and delivering structural sperm tail proteins in the basal body region, and adding the importance of understanding of the protein transport mechanisms. And also, once the sperm tail accessory structures, including the fibrous sheet and the outer dense fibers, form after the axon is established. Initially, the first and foremost thing that is going to establish in the developing spermatozoa will be axoneme. Surrounding the axoneme, there will be a fibrous sheet, and surrounding the fibrous sheet, there will be outer dense fiber, and surrounding the outer dense fiber, there will be a cilia membrane. So this FS structuring along with the principal piece and outer dense fiber development around the axoneme contributes to the overall, as I said, it contributes to the overall structural organization of the sperm tail. Similarly, like where we get to see this mitochondria here. So the mitochondria assemble basically this helically around the outer dense fiber during the final stages of spermiogenesis, particularly in the midpiece of the sperm tail. Here you get to see, right? Sorry. Yeah, here. So this is like you get to see during basically in the midpiece of the sperm tail, which assembles at the final stages of the spermiogenesis. And you get to see dynamics in the axoneme provide a very essential motor force for the sperm tail motility and also critical aspect for successful fertilization capacity. And also accessory structures, including the fiber sheet and as well as ODF, play a very important role in stabilizing the long axoneme, providing support to the entire tail for the movement, as well as contributing for the metabolic pathways and energy production pertaining to the motility. And this coordinated interplay of this molecular processes like microtubule structure and accessory elements like FS as well as ODF is crucial for efficient fertilization capacity of the sperm and during the maturation, that is spermiogenesis. So if you look at the proteome of this sperm tail formation, there are over 1,000 proteins which are associated with human sperm tail structures, revealing the complexity of it's very highly complex procedure, because if you look at the entire spermatogenesis, and around 2,000 plus proteins are involved in formation of spermatozoa, but 1,000 proteins alone are involved with the human tail structure, like formation of human tail structure. And the this extensive data provides insights into a potential as well as causative genes for acinospermia, a condition which is marked by reduced sperm motility. And also, out of this 1,000 proteins, 26% of this identified proteins relate to, very particular to metabolism, especially lipid metabolism, and suggesting fatty acid serves as the energy for substrate, which is very much important for sperm motility. And also, like proteins involved in sperm component formation and transport point to their role in male infertility also offers a lot of potential targets for research as well as therapeutic interventions. So as I said, yeah, if you look at it, initially the axoneme is formed at very early stages of the capping, the mature Golgi phase and cap phase, and followed by the fibrous sheet formation, which is again followed by the outer dense fibers, that is ODF formation. Of course, mentioned starts very after the axoneme is formed, and finally the mitochondrial sheet is formed. Here, if you look at it, the mitochondrial sheet remains outside, and the outer dense fibers remains just below the mitochondrial sheet, which is outer dense fiber again surrounds the fibrous sheet, that is FS. And if you look at the axoneme, which has a very specific or typical microtubule arrangement, that is 9+2. And if you look at the different chaperones as well as axonomal docking, a lot of proteins show different folds of activity throughout the spermiogenesis. And yeah, if you look at the ultrastructure of the mammalian spermatozoa, the flagellum, it is highly conserved and is divided into basically 4 parts. One is the connecting piece, the mid piece, and the principal piece, as well as the end piece. And this axoneme extends from the remnant of the centriole attached to the neck region to the implantation of the nucleus, that is the neck region, to the core structure along the length of the sperm tail. It starts from the neck region and ends in the terminal region of the tail. And axoneme is surrounded by, as I said, it is surrounded by these accessory structures like ODF and FS, and as well as MS. And this connecting piece contains a basal body that connects head and the tail together. And also this mid piece contains all the mitochondria found in the sperm and 9 ODF surrounding the axoneme. And in the principal piece of the sperm tail, these two ODFs are replaced by longitudinal columns of fibrous sheets, which are connected to each other by transverse ribs. And basically, the end piece of the sperm tail contains only the axonium surrounded by the plasma membrane. So here you don't get to see a lot of fibrous sheets as well as outer dense fibers. Here, the axon at the end piece of the spermatozoa, it is constituted of only the axonium, which is surrounded by the cilia membrane. And yeah, as I said, there are a lot of proteins which are involved. For example, with respect to mid piece, a lot of mitochondrial sheet, a lot of proteins which are well established have a huge and important, crucial, very important role at the time of formation of this mitochondrial sheet. So to name some, KLC3 to TGF2 to UBR, PP1, a lot more. And there are certain group of homobox genes of proteins which are also involved here. And also with respect to fibrous sheet, you get to see again different set of proteins like which are not repeated with the mitochondrial sheet, but with very specific to the fibrous sheet formation, which are involved in and the fibrous sheet surrounding the axonium. And similarly, this region which is surrounding the fibrous sheet, that is ODF, that is outer dense fibers. Again, you get to see different sets of proteins which have a unique role to play during the formation of this outer dense fiber during the spermiogenesis. And yeah, as I said earlier, this protein docking, this protein classification categorization basically happens in two different categories. That is, one is IMT, intramenstruate transport, as well as intraflagellar transport. So those proteins which are allotted to the IFT category will have a huge role to play in axonome assembly. That is the core structure where you get to see 9+2 arrangements of these microtubules is because of these proteins. And also the motility, the dynein arms which are involved in the motility is because of these proteins which are assembled at the IFT region, axonome assembly region. And also those proteins which are categorized to IMT, that is intramenstruate transport, they have a role, huge role to play in accessory structures like basically the sperm tail compartmentalization and the annulus formation, and and the connection of the head and the connecting piece, and submitting for the wave formation of this accessory structures like ODF and FS, and also mainly for the energy production for motility and also the hyperactivation of the spermatozoa. And if you look at this genes, main genes that are associated with the this 9+2 structure, here if you look at it, this connecting central bridge which connects the central microtubules with the surrounding 9 microtubules, there are different set of proteins which different set of genes which are involved, very much dedicated to this particular region, like RSPH1, RSPH4A, and a lot more genes. And if you look at this microtubule B and microtubule A, there are specific set of proteins like these are the microtubules alpha and as well as beta, and which has extended arm, like there is called as outer dynein arm, which is again assembled by a different set of proteins like DNH5, DNH8, and so on. And similarly, the inner dynein arms, which are marked at yellow color here, which has a different set of proteins to play a role. Except certain proteins has a overlap here, like DNH1, which are very common with respect to outer dynein arm as well as the inner dynein arm. And there are like radial spoke stack. If you look at the radial spoke stack here, it has a different set of genes which are involved. And the radial spoke stacks, if you see here, this spoke heads, what you get to see is different sets of proteins which are involved here. And well, yeah, increasing this male infertility is linked to, as we discussed in the earlier slide, a lot of inherited genetic variations, particularly mutations in the genes, which are very much crucial for the tail formation and also leading for the motility defects. And understanding this intricate protein networks involved in sperm differentiation is crucial for addressing male infertility and also identifying the potential therapeutic targets. As I said, yes, sperm motility is driven by the activation of the flagellum, relies on the axonium, a core structure resemble motile cilia, and this genetic mutations affecting this axonium formation contribute to again, male infertility. Because I'm not going into the detail because we have a lined up lecture by Sayali and lined up a panel which we're going to discuss in detail about. So I'm just going to discuss about the anatomy and the mechanisms that regulate the sperm tail, as well as the motility in the sperm, as well as hyperactivation in the sperm tail. And they're going to deal what happens if there is abnormal sperm tail formation. What are the consequences of this abnormal sperm tail formation? So, and also beyond this axoneme, various accessory structures are essential for maintaining the sperm tail's integrity, like facilitating the capacitation process, and also generating the energy during the sperm's journey to fertilize the oocyte. Well, with respect to coming to the mechanisms of sperm motility, if you look at the mechanism of sperm motility, yeah, it relies basically on different metabolic pathways as well as regulatory mechanisms, which are crucial for the sperm to attain motility in order to achieve successful fertilization. So basically, there are two broad categories of pathways which are very much crucial for initiating the sperm motility in the spermatozoa. One is the calcium pathway, as well as second is the cyclin adenosine monophosphate dependent protein kinase, or PKA pathway, which plays a huge role in regulating sperm motility. So these pathways involve a lot of subcomponents like calcium ions, adenyl cyclases, bicarbonate ions, membrane channels, as well as the process called as phosphorylation. And all these are needed in order to make the spermatozoa achieve the capacitation process, hyperactivation, as well as actosome reaction. And beyond the specific gene defects, any abnormalities in these metabolic pathways and regulatory mechanisms can lead to a very poor sperm motility, becoming a potential cause for infertility. So understanding these and addressing these factors is very much important with respect to any interventions that you get to do in infertility. Excuse me. So if you look at the mechanism, how it activates, activation of this sodium, how this sperm motility is triggered. Initially, what happens is the activation of this sodium plus ions and bicarbonate co-transporter and the regulation of this SLC26 transporters increase the bicarbonate levels in the cytoplasm. So once this activated, so this activation of the sperm, once bicarbonate levels are increased here, this activation of the sperm, this will help in activation the another very specific receptor called as SNHE, which is a sodium proton exchanger, which is aligned with the activation of this proton channel, will lead to the increase in the internal pH, which in turn, once the internal pH is increased, that will activate the CatSper, which is a cation channel of sperm that enables the entry of calcium ions and thus increases the internal calcium ion concentration in the cytoplasm. And if you look at here, along with that, there is also like a hormones. If you look at hormones, progesterone, which is a steroid hormone synthesized by the cumulus or granulosa cells, have a huge important role in activating the CatSper, either by binding to the channel itself or an associated protein with respect to CatSper. And also albumin has an important role to play here, which the main protein of the human blood plasma and an oocyte glycoprotein also helps in alkalization of the sperm cytoplasm that elevates the internal calcium ion concentration. So basically the overall calcium ion concentration increases with the moment the internal calcium ion concentration increases, that induces or that influences glycolysis to and also the axoneme activity, promoting the hyperactivation of the motility. So, and furthermore, there are like this bicarbonate as well as calcium ions regulate a typical soluble adenyl cyclase, which generates cyclic AMP, cyclic adenosine monophosphate, and that in turn activates protein kinase A, which has a huge role to play here, because this PK induces phosphorylation of this axonal dynein. Remember this increase in bicarbonate as well as calcium ions regulate the typical soluble. This will activate this atypical soluble adenyl cyclase, which generates the ATP to cyclic AMP, which in turn activates the protein kinase A, PKA. So this PKA induces phosphorylation of this axonal dynein, leading to the consumption of ATP, and thus increases the intercellular pH. So finally, this PK activates sperm tyrosine kinases with very specific regions like serine and phosphorylating on very specific regions like serine as well as threonine residues, and also to get a cascade of proteins which are involved in phosphorylation which finally helps in getting the sperm motility. So also this increased cyclic AMP may activate this PKA and turn inactivates. See, if you look at here, this cyclic AMP in turn, along with increased pH, along with this increased calcium ion concentration, will lead to hyperactivation. That is, which is very much one of the important behavior of sperm motility before fertilization. With respect to, this is with respect to triggering the motility in case of human spermatozoa. With respect to the mechanisms that trigger the sperm capacitation, or if you consider this as external environment and this as flagellum, and this increase in protein tyrosine phosphorylation during capacitation is regulated by cyclic AMP dependent pathway, which involves a PKA receptor. There are two types of activators, like one is the protein receptor tyrosine kinase receptor based pathway and non-tyrosine kinase receptor based pathway. So initially we'll talk about receptor based pathway, where this cyclic AMPs, once they're activated, they won't bind on a family of proteins like AKP family of proteins, which indirectly activates this tyrosine kinase, and in turn, this tyrosine kinases will lead to phosphorylation of this dynein-rich regions, which will end up in the capacitation of spermatozoa. Yeah, this is something similar. There are certain proteins, namely AKP53 as well as FSP95, which are prominent tyrosine phosphorylated proteins during capacitation. And basically, with contrast to that receptor-based pathway, non-receptor-based pathway has a transmembrane domain, which is mostly a soluble intracellular protein located on the gel on the cytoplasm nucleus, as well as which is basically anchored in the inner leaflet of the plasma membrane. So basically, this tyrosine as well as protein phosphorylation of the sperm flagella proteins leads to capacitation of the human spermatozoa. Yeah, recently we also discovered how the sperm moves. Like initially we thought it is going like it is moving side by side, but now with the help of this 3D microscopy study, which overturns the hundreds of years of reproductive science. Let's go through a small journey of this video. To just one side, but rather two sides. Also, you have to protect the avoid from the circles. This one side. Yeah, so this was a recent discovery where we thought sperm moves side by side, but initially with the advent of this 3D microscopy, we get to know that it's like corkscrew-like structure, like a movement, what the spermatozoa actually does to progress forward. And coming to the take-home message with respect to centrioles, dual centrioles in the spermatozoa, as I discussed earlier, like it has two centrioles, one canonical as well as one atypical, highlighting the very unique structural composition in comparison to other cilias. And it has a very critical function in spermatogenesis, like where it plays a very important role in spermatogenesis, contributing to the cell divisions and also the formation of the flagellum, and also important, like underscoring the importance of the development of the mature spermatozoa. With respect to the beyond spermatogenesis, the centrioles in mature spermatozoa have essential functions like linking the head to the tail, as well as regulating the beating of the flagellum, as we just saw in the previous video, and also which is very important for the sperm motility. So it's just not restricted to the cell division as well as spermatogenesis and the sperm formation. It has the role to play even post fertilization, where the sperm centrioles continue to exert influence post-fertilization by actively participating in the shaping the cytoskeleton of the zygote, contributing to the early embryonic development. As I said in the earlier slide, I showed the formation of ashters, which is in the human, at least in humans, it is from the centrioles that is contributed from the sperm. And yeah, it also have a different distinctive roles to play, like along with these above roles. Coming to the take-home message of the sperm tail, the mammalian sperm tail exhibits highly conserved ultrastructure comprising of 4 distinct parts and highlighting the very complex organization. And the axoneme extending from the centriole remnant serves as a core structure along the entire length of the sperm tail, starting from the neck to the tail. Axoneme will remain the same, emphasizing that emphasizes its very important role in the sperm motility. And also this accessory structures like outer dense fibers as well as fiber sheet and as well as the mitochondrial sheet contributes for the overall structural complexity of the sperm tail. And also this this interplay of this different components like calcium ions, cyclases, bicarbonate ions, and phosphorylation events will help the sperm to attain capacitation, hyperactivity, and also acrosome reaction, which is very much crucial for the fertilization process. So overall, if you look at it with respect to sperm neck, that is centrioles and the mitochondria as well as the tail region. So if you get to see the sperm head and tail, the structural sperm features are very much important. And with respect to the morpho-functional sperm structures, which is with respect to motility, abnormalities, and also oxidative stress, they are also important, along with intrinsic sperm features like sperm DNA, proteins, organelles, and microRNAs and non-coding RNAs. Combining of all this will lead to a healthy, viable spermatozoa, which is important. So again, yeah, I would like to quote my talk with this statement. See, initially I wrote this without knowing this importance of the amount of proteins and structures that are involved in acrosome as well as midpiece as well as tail. I mean, neck region as well as the tail region. So ICSI is like injecting 3 million DNA base pairs and additional 50 to 100 sets of mitochondrial DNA, centrioles and epigenetic marks, microRNAs, proteins, and above all, a big hope of fertilization, along with those acrosomal, the cargo, what we get to see in the acrosome, and the cargo, what we get to see in the tail. Thank you. And my talk here.
Thank you so much, sir, for this detailed talk, sir. And there are so many questions right now with us, but I would just stick to the question related to our topic. So if there's a very simple question I would like to ask by the students here is how much effect of the sperm middle piece and tail morphology is seen on the embryo development?
Yes, I just mentioned that, right? Like if you, first of all, you should have a motility. If there is no motility, then you have to do a viability test and you have to check whether you have to induce the motility with respect to, with the help of theophylline-based products to whether to see the sperm is viable or not. So basically, this structure, which is pertaining to the neck region as well as tail, midpiece and as well as tail region, helps us to identify whether the sperm is viable or not because of the motility it is going to form, it is going to exert. And secondly, the centrioles, as I mentioned in the slides, the sperm is going to contribute the centrioles, which is important for the earlier embryonic divisions.
So similar question is there, like as you have answered that if the person is having no normal sperm, then what criteria do you use while choosing a sperm for ICSI? And if it is globospermia, what do you use? ICSI, IMSI, what is the preference and what is the data saying, sir? This is the question by the audience.
I think this question, I think we will take in panel because they are doing panel on morphology only.
Sir.
So I think Sanket, it was wonderful lecture. Actually, I got so many insight of this things, tail, neck, and one thing from my side.
Yes, sir.
It is always in my mind actually, or I think most of our delegates participants want to know that what happened after the fertilization with regards to tail and neck. Because after fertilization, this is always a question in the mind. If you are doing ICSI, then what is the need of that tail and neck? The thing is always in mind. I think most of people want to know that thing. Just tell us about this.
So what happens in vivo, we don't know actually whether the entire tail is engulfed into the cytoplasm or not. But at least with respect to ICSI, what I'm trying to say is here is this: the moment you do ICSI, we are injecting the entire the spermatozoa inside the cytoplasm. So once there is like additional formation of this pronucleus, before the formation of pronucleus from the male end and female end, this mitochondria which is contributed from the male will be like they'll be like destroyed and they'll be thrown out. Mostly 99.9% times. I've seen some case reports where I've seen a Chinese male which shares like mitochondria from father as well as mother both. And there are some hardly 17 case reports which have been published where they share heteroplasmic kind of condition where the mitochondria is contributed from the father as well as the mother. Otherwise, in nature, this mitochondria are thrown out. And with respect to this axoneme structures, the dyneins, microtubules and all, yes, again, this will be like destroyed with respect to ubiquitination, I don't know, ubiquitination-based pathways, which will be not used for early development of an embryo.
Thank you. Thank you. Anything, Dr. Sanjay is there. I think he has something in mind. Dr. Sanjay, are you there? Driving na kar rahe ho.
I think Sanjay sir just now got disconnected. So we will carry forward, sir. We will take up the question in panel now.
Sure.
Thank you so much, sir, for the wonderful presentation. And we'll invite the next speaker, sir.
Sanjay, you just be there. Sanjay, be there for panel, na? We will discuss in that panel also.
Okay.
Yes. Yeah. I think, Nidhi, we have a polling.
Yes, sir. So for all the participants and our viewers, we have arranged a small polling session in which we will be displaying 5 questions right now. And with each question, we will be giving you around about 20 seconds. And then we will be declaring the answer simultaneously to brush up our knowledge. So let's start with the polling. Can I have the first question?
Yes, ma'am.
The first question is dash molecular motor for sperm movement. The choices are axoneme, flagella, dynein, and cilia. Please submit your answers.
Those who have heard my lecture would like it will be easy for them to answer the question, this question at least.
I think we cannot participate, na?
No, sir, it's for the audience. Can we see the poll now?
Yes. These are the results.
Okay. Sanket sir would like to take over the poll questions.
Yeah, sure. So, next question, please.
Yes, sir.
So the next question would be, if you can suspect dash in a male with 100% immotile spermatozoa with normal vitality. So the options would be there will be a single choice from the options. Like it will it be a Kalman syndrome, Carter-Gunner syndrome, Young syndrome, as well as Klinefelter syndrome. Last 5 seconds left. So 33% of them answered it as the Kalman syndrome, and 67% has answered as Carter-Gunner syndrome. Well, coming to the next third question. Can you have the question, please?
Yes, sir.
Well, the presence of cytoplasmic droplet in the mature spermatozoa could be due to defect in dash spermiation. Option A is spermiation. Option B is spermiogenesis. Option C is initial part of spermatogenesis. Option D is ejaculation. Poll has ended. And the answers would be like 33% of them again voted for spermiation and 67% of them voted for spermiogenesis. Well, coming to the next question. In asthenospermia, you can expect semen report with more dash defects. Head defects, option B, neck defects, option C, midpiece defects, option D, tail defects. Yeah, now this time 33% of them voted for midpiece defects and 67% of them voted for tail defects. Well, coming to the last question, fifth question. Is it possible to do ICSI in semen sample with 100% immaterial spermatozoa? If yes, how? And options would be yes, eosin agricin stain. Option B would be no, because all are dead. Option C would be yes, HOS test, hypoosmotic swelling test. And option D, no, because all are immotile. Do we have Ananta here to reveal the answers?
Actually, Ananta is traveling.
Okay, sir. Answers are with the technical team, right, sir? So
Yeah.
most of the people have answered it rightly, like yes, it is a hypoosmotic swelling test. So what we're gonna do if we find 100% immotile spermatozoa in the semen sample before ICSI. So who is going to display the answers, sir?
So I have the answers with me. So are we showing it on the poll right now? So the best part is about 100% participation in the last answer, sir. Then absolutely correct, sir. It is with the help of horse test we can see, we can do the ICSI. So should I carry forward, sir?
Yeah.
Just reveal the other four answers, no?
So the first question, the question was dash molecular motor for sperm motor movement. The answer is dynein. Then the second question was, you can suspect dash in a male with 100% immotile spermatozoa. It was mainly the Cartagena syndrome, in which it occurs. The third was presence of cytoplasmic droplet in the mature spermatozoa could be due to defect in dash. So the answer was spermiogenesis. And the fourth question was, in asthenospermia, you can expect semen report with more dash defects. So the answer was more of tail defects. And I would again like to thank Dr. Ananta for making this wonderful polling questions. So taking forth, so I request our next speaker. I invite our next speaker, Dr. Siali. Wait, sir. Could you please introduce, ma'am?
Nidhi.
Yes, sir.
I think there is something which probably I would like to ask to Sanket.
Okay, sir.
Yes, sir.
It is regarding the usage of motility enhancers like caffeine or the platelet activating factors or the derivatives of pentoxyphylline or etophylline. So can you put some light on that? Because you mentioned only about HOS, but these agents are also being used in cases of very poor motility or absolutely no motility.
Sir, yeah, to answer your question, yeah, as you rightly mentioned, yes, these are also used as adjuvants at time of when you get to see a lot of humoral spermatozoa, like almost 100% humoral spermatozoa, or with respect to ejaculated spermatozoa or testicular spermatozoa. So basically, how this going to regulate this motility part is, as I mentioned, the two pathways which are like protein kinase dependent pathway as well as the PK pathway, where this inflow of the sodium as well as calcium ions, which will in turn increase the activate the Caspar ion channel. And once this Caspar ion channel is activated, a lot of calcium are getting into the cytoplasm, the external source, which in turn will increase the glycolysis of the at the mitochondria at the region of axoneme. So in turn, this will enhance the motility. So probably this is a hypothetical thing what I got to know after reading about this, sir. But in detail, there are a lot of other mechanisms which are involved here, like parallel to this, there are other things which also involved, which will enhance the motility in case of 100% hematological spermatozoa.
Perfect. Thank you.
Thank you, sir.
Thank you so much, sir, again. And I invite Siali ma'am. Ma'am has done an MSc PhD in reproductive biology. Is it visible? The CV is visible?
Yeah, it's visible.
And
Yes,
regenerative medicine. Good evening, ma'am. From the Northwestern University, Chicago, USA. She is PG CRT reproductive genetics from MUHS, India. Ma'am has a certification in clinical embryology from Cleveland Clinic, USA. Ma'am has done she is an ASRM certified clinical embryologist, and PGT PGS embryo biopsy certification has been done from Spain. Ma'am is again fertility preservation PGCRT done from ASRM. Ma'am has microbiome therapeutics PGCRTs done from Stanford University, and also clinical research PGCRT from certification from John Hopkins University. Welcome, ma'am. Ma'am is currently the CEO and founder of Cell Shore Biotech Research Center, India, and the clinical and scientific director of Aruti IVF LLP, India. Welcome, ma'am.
Thank you. Thank you for the nice welcome. Sorry, I have a sore throat, so I will try to keep the presentation to the point. I'll start sharing my screen. Oh, just give me a minute. Is it visible?
Yes, ma'am.
All right. Okay. So today, the topic given to me after coming in after Sanket is the effect of head and neck, basically neck and tail defects. But I'm going to cover in general sperm morphology because, of course, there are different proteins looking at just the head, just the neck, but we want to look clinically as to whether any of this selection in any of this is going to improve results. So, having said that, we already have discussed how normal sperm morphology looks like and how it evolved from the time of Leeuwenhoek. So I won't be looking at it again. Although, as you know, this image that you have all seen in the WHO guidelines is the primary teaching method for us to learn what is normal versus abnormal spermatozoa. And I believe we should keep updating us from time to time as to what it looks like, because after you turn a little bit senior, you tend to not be doing semen analysis more often. But I believe that is a cornerstone of a good embryologist and andrologist. But one of few of the clinical issues that occur, and they have a name, is uh, the defects that are shown on the screen: globozoospermia, where you have a lack of acrosome; pinheads, where you may or may not have the midpiece; acephalia versus complete pinheads, having genetic factors involved, microcephaly where the sperm heads are normal, and macro where they are bigger. Either of them is going to really impair the spermatozoa and affect your outcomes. Now, currently at this point of time, we almost all of us refer to the strict Kruger strict criteria, and we have all known that 4% is the cutoff. What we tend to forget is 4 to 14% does have a slight decrease in fertility, and over 14% is what is considered completely normal, or ability to fertilize naturally without any impairment. Absolutely below 4%, you have teratozospermia, and you have concomitant issues with, of course, you cannot do either IVF, and even IUIs is not recommended. Even IVF is something that you would not take a risk with, and we will look at what are the modalities if you have teratozospermia and whether it has any effect on outcomes. And what really affects sperm morphology, I think Sanket has probably covered what all problems we could have and which proteins and genes are involved. However, a lot of it could be genetic, but many of the factors are environmental in origin. Many of the factors could be disease states like cancer, the induced states like smoking, ROS caused by varicocele, and other infectious agents that could definitely lead to more morphological defects in the spermatozoa. Because if you have any of these interfering in the spermiation stage or spermatogenesis stage or the maturation stage, you're going to see morphological defects. This is a very simple slide. What are you what are you allowing the spermatozoa to do? How much maturation is allowed to undergo? It depends on what is the method that you're utilizing in your ART practice. And coming to the points clinically as to how do you measure abnormalities? Apart from doing morphology via staining, we have something called the teratozospermia index, and I believe this should be utilized more by our faculty. I believe the penetration is not that high. There are a lot of CASA machines that do give a TZI index, but it is very well possible to do it in wet manual semen analysis. It's a very simple method to do it. It's total number of abnormalities in each category upon total number of abnormal spermatozoa counted, but this is if you do staining for morphology, and it is always advisable to do for a semen analysis. And TZI that is above 1 point associated with post-sperm fertilizing ability, and it is currently we do not know how much of an effect it has. It is always preferable to mention it per patient so you can decide what modality you're using. So, what are the effects of the sperm defects on ICSI outcomes? And what exactly should we be looking at? So, at this point of time in ART practice, we are looking at sperm DNA fragmentation that may or may not be correlated to morphology, and we are also looking at morphological parameters. We know that abnormal sperm morphology is associated with lower rates all across fertilization, cleavage, embryo development, lower implantation. We have a lower number of embryos formed in teratozospermic patients. There is also correlation with nuclear decondensation, which is correlated to vacuoles. I think that was one thing that Sanket may or may not have touched upon, that vacuoles, big vacuoles are correlated to the disability of nuclear decondensation to happen, which is the basic principle behind IMSI, and that is the reason why IMSI is utilized in such samples. So these are the main correlations: high sperm DNA fragmentation and low morphological index is responsible for lower outcomes. What current modalities do we have? And not all of these are for selecting morphology. To be very honest, I think morphology has been a factor that has not really received the attention that it should. And we have mainly been looking at the functional biomarkers and the biomarker based selection criteria. We do have when you look at the clinical algorithm of how we treat the patient, we look at the WHO 2010 SFA or HSA, semen analysis along with morphology. We sometimes do spontaneous fragmentation, but it's very canonical as to where it will be utilized. We have biomarkers like hyaluronic acid, Pixie, or we have microfluidics in some cases, or Max for reducing, removing the apoptic cells, apoptic sperm cells. However, the correlation to morphology, I believe, has not been studied that extensively as it should be. And we'll be looking at a few of the studies that have really looked at morphology and the ultrastructural abnormalities as well as electron microscopy images to evaluate what is the effect on and what outcome that particular patient has after ICSI. And like you can see on the screen, we have had a discussion of centrine and axonomal and perioxonomal alterations, and how the differences in the centrioles could affect fertilization and eventual formation of embryos. But here you have proof that this indeed in some cases does not lead to embryo formation. So, for example, in the first, if you see that this with the case studies, and I believe case studies play a huge role in understanding in the context of customized treatment as to how really you can look at the patient and what that particular patient has in terms of treating them. So, in that first patient, you had incomplete axonome cross sections and weak tubulin signaling. Centrin 1 was normal. ICSI, after ICSI, a healthy baby was born. So really nothing was required, maybe for them. And in the second patient, they had reduced tail length. Now this is something that I don't think we will be able to really see in ICSI or even in MC. We would not be measuring the tail length, but yeah, the reduced tail length, axonomal and periaxonomal alterations, the fragility of head-tail junction. I think all embryologists here and people who actually do micromanipulation would have encountered some cases where the sperm are very fragile. And when we mean fragile, they on immobilization, they immediately separate, their head and neck separates, and that is a pathological condition that is caused sometimes by genetic factors. It could be caused because of bacterial infection. It could be also caused due to osmolality issues that you have created heterogeneically in the lab, definitely. But in some cases, and in many cases which are unexplained repeated failures with such parameters, they could have a genetic origin. And here, in this case, centrin 1 was altered in 80% of sperm and no embryos were formed. In the third patient, tails were reduced in length and they had a disruption of the fibrous sheath, which they could see under ultramicroscopy. And two embryos were transferred, no pregnancy occurred. The point that we are trying to make with these studies is that there are genetic factors in some patients that could lead to this morphological defect. So be on a lookout as to how these sperm behave under micromanipulation. Do not disregard it as maybe we didn't, process it properly, or maybe the osmolality has changed. It could potentially be a patient issue. And yes, and this was something that's very interesting in patients with acephalic spermatozoa. So acephaly is basically head is absent, but you do have mid pieces, and these are called pinheads by mistake. They are not really pinheads; they're acephalic. And when they're acephalic, they tend to have a genetic origin, and in that there are three subtypes that were studied. And in some, especially in subtype 2, when these three genes were disturbed, you still could end up getting clinical pregnancies and live births, which is astonishing to us. Astonishing because you don't really see an acrosome, but they do contain apparently enough genetic material to lead to live births. But these are acephalic spermatozoa. They lack the head, but they do contain the midpiece and a part of the head. So that was certain sperm abnormalities. Now there is one study that I'm sure a lot of you have seen, but would come as a surprise to some of you, is 282 retrospective. I'm sorry. a retrospective study was done in more than 200 cycles, and they selected only abnormal spermatozoa in the control group, and in the treatment group normal spermatozoa. I really pity the embryologists who actually did this study, because they had to do ICSI with abnormal spermatozoa, and I would really believe how tense they would be in doing that. But they did not have any difference in as such the or normal morphology of the embryos. However, implantation rate was severely low. I'm sorry. Excuse me. Sorry for that. I'm not well. Just give me a minute. I'm sorry. Can you see? Nidhi, can you see the screen?
Yes, please continue.
Yes, sorry. All right. So, and the next study talks about how flagellar abnormalities are causing slower embryo cleavage kinetics in patients and can lead to lower pregnancy rates. In such patients who had flagellar defects and severely impaired spermatozoa, still 45% of men could father children. So I think it is very important to know that even if the patient has a high teratozoospermia index or has severe teratozoospermia, they still have a good capacity of having healthy children. So these are the different options that we have for poor sperm morphology. We have ICSI, which is used by all of us, IMSI, which has been utilized in a lot of cases, PIXI, that is utilized when the hyaluronan biomarker is impaired, and artificial oocyte activation in some cases where calcium ionophore defects are thought about. So in the screen, we see there are so many modalities. However, not all of them will select for good morphology sperm. The most utilized one is density gradient and swim up. Density gradient really does not improve DFI integrity, but it does select sperm with normal morphology more, because as we know, the whole principle of density is based on the density being different of morphologically normal versus abnormal sperm. So it is definitely what do you say, the gold standard for sperm selection or sperm processing for any cycle in IVF or ICSI. However, it does not have any advantages over swim up in mild infertility, and in high TZI index, it is always suggested to do DGC plus swim. Apoptotic sperm deselection max has been shown to reduce DNA fragmentation index. No correlation has been shown with morphology. Microfluidics, again, so much work has been done on reducing DNA fragmentation via microfluidics. We don't really know if it improves the morphology. However, whenever it has been compared with density gradient, and our own work on positive rheotaxis also shows this, it has a very similar enrichment in morphology as that of density gradient, which in itself is a good prognosis for utilizing it as a single technique for uh, processing spermatozoa. And HF binding has not really been shown conclusive in a large randomized trial. However, there have been some reports even after the trial that talk about indicated cases, especially with lower hyaluronan binding, especially with teratozoosphermia, could be benefited by PIXI. But all of this is conjecture, all of this is under investigation, and we really should be documenting our case studies to know that how really we are utilizing this technology for our patients. IMSI, I kept IMSI last because I think IMSI was quite promising when it came in. We used it quite a lot for about 12 years. We still use it from time to time, but we don't use it for every patient. I'll also just briefly talk about what our current clinical work, research work is about, utilizing strict criteria. However, the Cochrane review had very few studies on IMSI. Very few studies on IMSI were done because the number of patients that qualify for IMSI according to strict criteria are less. And even in that, there's no randomized control trial done in the Cochrane review, and it was inconclusive. However, to continue, this is all IMSI data. And this is from this very beautiful review called Tales of the Tail, and the name of this webinar is based on that paper. I feel Tales of the Tail and the Sperm Headaches. That's what it's called. It's a very nice review paper, and it looks at IMSI versus ICSI for nuclear vacuoles or defective chromatin condensation, as we call it. And basically the concept is that the larger the nuclear vacuole that's observed, more than 30% of the volume of the head, of the complete head, that would impair your results, and that is where IMSI is suggested. And as you can see here, there are quite large differences in implantation rate and pregnancy rate and miscarriage rates in the use of IMSI in such indicated cases. So I still think we should not be dismissing the particular procedures, but we really should be looking at where we are utilizing them. And this is particularly about head neck abnormalities and outcomes. And when I say head neck abnormalities, the one at the top of the mind is globozoospermia. And acrosomal hypoplasia is basically a biomechanical lack of elasticity in the head and neck region, which could potentially lead to lack of forward kinetic motility, lower velocity. And since not all of us have the method to detect whether they have lower velocity, and semen analysis does not always correlate to process samples, ICSI samples, sometimes you have to evaluate this on process spermatozoa. As you can see, however, globozoospermia is pretty clear to know, and in globozoospermia, the utilization of ICSI is with good results actually. If you use ICSI with or without calcium ionophore, there have been live births reported. With calcium ionophore, the possibility of forming embryos is higher, and that has been shown in the retrospective studies. And immotile spermatozoa. So we spoke about immotile spermatozoa and why they occur. We also talked about some genetic disorders where they occur, PCD and Carter-Gunner syndrome. In PCD mean primary ciliary dyskinesia, kinesia, as you can see on the screen. In this case, when you utilize horse or other modalities like pentoxifillin, and I think this is a combination of all three. There are a lot of studies in this, and they have pooled the results. Live births were obtained, either with using non-contact diode, which is laser, or using the theophylline or pentoxifylline for as a motility inducer, or using HOS. But I believe in the last 10 years or so, we have really moved towards using motility inducers, and those are available in the market and with good outcomes. So HOS really has lost its primary, a gold standard for immortality, and uh, there are quite good results that are being shown. Of course, miscarriage rates are reported, but very similar to normal ICSI outcomes. You have good amount of live births to say that this is a good modality used for immotile spermatozoa. And like I said, whatever you utilize for morphology, before you even start doing, looking at the effect of morphology on IVF ICSI cases in your own setup, you first need to have some quality control in your own andrology laboratory, and try to send your semen analysis and morphology testing to a reputed laboratory who know what they are doing. Especially who have learned and are trained in WHO guidelines for semen analysis. And that is when you are going to have reproducible results for morphology, have teaser eye index documented for every case, and just control for any escalation that happens in reduced outcomes in your cases. And some of the research work that we have done on uh, sperm selection is we have used positive rheotaxis for more than 5 years. We have been working on this in around 7 centers in India, and we have based this work on D. Martin's work. This was the work that we had first done, and we had a very respectable clinical pregnancy achieved in all field cycles in the first ICSI. So this was a cross-sectional retrospective study, and we achieved 42% clinical pregnancy rates with positive rheotaxis. We then shifted and moved on this work to the prospective 2-arm pilot study looking at Bologna criteria 4, low prognosis or poor ovarian reserve patients, where we were looking at trying to utilize a standard sperm selection method that does not add costs to the patient, not an add-on, but just replace the crude ICSI for all cases of corresponders. And we were able to do this with this particular method, and our results, we alternated it for every cycle and randomize it every cycle. These were the results. We had a very respectable pregnancy rate. Conventional ICSI had 45%, PRD ICSI had 58. Our day 3 good quality embryos were higher with PRD. Of course, the numbers are low, but we are still continuing the work, and the blastocyst rates were also very respectably improved. And we had lower cycle cancellations, which I think is clinically very important when you are dealing with cases. Yeah, so now we are going to focus on using PRED. So we have developed PRED strict, and we are focused on using this for patients with a higher TZI index, and we'll keep you posted as to what happens. And thank you for your time, and thank you for this opportunity. And sorry that I was not well. Thank you.
Thank you so much, ma'am. And Ved sir, you have any questions?
There is some questions in that. Let me check. Yeah.
The question I can see here is sperm quality affecting the tropoectoderms?
Right,
Oh.
right.
Wow.
Very difficult question.
Yeah, I think what we would have to do to know that is so much work, because we will have to first see which trophectoderm markers are really taken from the male gamete. We don't really know whether any of that, but I do believe that if you don't have blastocysts, then yes, trophectoderm is affected. So I think it's correlated to how many embryos really go to blastocyst stage. I don't know if it can be directly correlated to the trophectoderm itself, but I do believe that any morphological issue that cannot be solved by ICSI is going to show its face somewhere in the embryonic cycle, because 50% of the DNA does come from here. And you have to understand that morphological defects means that there could be a defect in how the DNA is transported into forming the aster. And if the aster formation is not proper, you're going to have issues with blastocyst formation. So I hope that answers the question.
There is one more question. If a person not have any normal sperm, then what criteria we should use to choose a sperm for ICSI? And related to that, if globospermia, so what we should do, ICSI or IMSI?
Oh, so to be very honest, if you have 0% normal morphology, the first thing that you need to look at is that whether you can do whether you can counsel the patient that this may or may not result in embryos, because you know that complete teratozoospermia does give babies. It is not a marker that is going to go completely. Acephaly gives babies. Can you believe this? Acephaly gives 45% of men with acephaly have children. So uh, even if you have 0% normal spermatozoa, please do not counsel your patient for sperm donation. Kindly, please give them an option of ICSI. If you really want, give them an add-on. Please use PRAD ICSI. It's free. It's free for everybody. Please, if you can use it, go on your 40x objective. If you do not have IMSI, use strict criteria while you are doing ICSI. I believe that solves a lot of the issues, and we really should be collecting this information as to what 40S ICSI is versus MC. And I don't think there is a huge difference.
Yeah, you're right.
Yeah.
Absolutely right.
So that's one thing. Sir, the other question was globozoospermia. Now, globozoospermia, you have to understand the problem. Now, globozoospermia is the acrosome is absent. Now, you are saying do IMSI. IMSI is for morphological selection. You are using the wrong tool for the wrong problem. When acrosome is absent, your problem is going to be a lack of calcium ionophore activation. It's going to be a lack of Caspar signaling. Um, that's the problem you should solve using calcium ionophore, not MC. If you do not have complete globozoospermia, then sure, you can use MC.
Yeah, just to extend what Sally has said, first thing is you have to rule out whether it's the complete globosospermia or partial globosospermia.
Yes.
Once you're done with it, yeah, then probably you can use the MC or other tools to enhance the fertility outcomes.
Any other questions?
I think there is no other question. Nidhi, thank you so much, Sally, for giving so good great lecture. And
thank you, ma'am.
sorry if you are not well, but thank you so much. Thank you.
Thank you for enlightening us. So we will start with the next panel discussion.
Before panel, I think there is poll.
No, there are 5 more questions for the poll.
Yeah.
Oh.
So I don't know why I always miss the
I
poll.
answered all 5 questions correctly. I was so scared I would not. And I'm so happy because I listened to Sanket's lecture. Thank you, Sanket.
Thank you, sir.
So changing the little pattern of the poll, we'll have all the questions together, and then in the end we will see the result of the poll and the answers together. So can we have the poll questions? Sanket sir, you'd like to start with the poll questions again?
Yeah, definitely. Please.
Yes. So please do. Can we have the poll questions?
Yes, ma'am.
No, I'm getting all the questions. Can you go one by one?
Sir, it is all the question together this time. Little change in the pattern, and then we will show
the
the
first
result.
question will be parts of sperm flagellum are. First option would be head, neck, and tail. Second option would be midpiece, principal piece, as well as the end piece. And third option will be midpiece and neck and tail. And fourth option would be head, midpiece, and tail. And the second question would be.
No, sir, we'll just take a gap of 15 seconds, sir.
You need to take a gap?
We continue? Okay. Just take 10 seconds gap with each question, and then we'll go forward.
Well, the second question would be dash differentiates mid-piece and the principal piece. Of course, this I covered in my lecture also. So guys, you have to answer it rightly. So first option would be axoneme, and the second would be the renin arm, and third will be annulus, and fourth would be neck. Dash differentiates mid-piece and the principal piece. I specifically mentioned in my lecture.
Third question, sir.
Yeah, well, coming to third questions, can we have the options, please? Yeah, DASH helps in organizing cytoskeleton of the zygote to mediate pronuclear migration and cell cleavage. Even this I have covered, I think, even at take-home message. Options, please. DASH helps in organizing cytoskeleton of the zygote to mediate pronuclear migration and cell cleavage. Technical team, can we have the options for the
The
third?
option is just I'll read the options. It's the head, axoneme, both head and axoneme, that is part A and part B, and the fourth option is centriole. The fourth question is, both proximal and distal centrioles are present in the middle piece. Is it true or is it false?
Both PC as well as DC are present in the mid piece.
Mid piece. Is it true or is it false? And the last question is question number 5. That is, centrioles are important for mitosis, B, meiosis, both mitosis and meiosis, and last is cell death. So please select your answer. Can we have the poll results?
Dr. Visha, yeah. The first question, most of them answered as mid piece, principal piece, and end piece. 67% of them, and only 33% of them answered as mid piece and neck and as well as tail.
So the 67% are actually correct. That the answer is mid piece, principal piece, and end piece.
Yes. Terminal piece. Well, the second question would be like dash differentiate mid piece and principal piece. It is differentiate mid piece and principal piece.
So everybody has written axoneme as an answer. The answer is annulus.
Yeah.
So there's somewhere the message has gone wrongly.
Sanket, you have to take the lecture again.
So the third question, that was, dash helps in organizing cytoskeleton of the zygote to mediate pronuclear migration and cell cleavage. So it's a 50-50 choice. That is part A and part B. And then 50% of them have said centriole. So the answer for this question is centrioles. Fourth question, both proximal and distal centriole are present in midpiece. That is true. Okay. And the last part was, centriole are important for dash. So just a minute. So again, it's a 50-50 choice, part B, and some people have said it is both mitosis and meiosis, and the answer is both. It is important for both, whether it is either mitosis or meiosis.
True. Thank you, Ananta. Thank you so much.
Any message from Siali ma'am and Sanket sir for our viewers? After the poll.
Oh my God. I think I should be brushing up on my andrology as well. It's not easy to answer basic biology questions now after so many years of clinical practice. But what I'm just looking forward to the panel because I believe it's going to be very interesting. And my last message would be, don't disregard methods that have been proven by RCTs to not be useful for all patients. Not all methods are useful for all patients, but just evaluate, focus more on andrology diagnostics and the quality of the report. That is missing in India in a lot of places. And that is where the important message would be. Thank you.
Yeah, right.
Thank you so much,
My
ma'am.
sort of first message would be, even though the sperms are present in millions, please don't take it for granted. A lot of
Yes,
things
sir.
that you have to consider. Well, over to you, Nidhi.
Thank you so much, sir. Thank you so much, ma'am. And actually, the main aim or the main aim by which IHERA team supports is the core study. Everybody is doing practical experiences. Everybody is having. So we are now targeting the core, the basics. That is the main aim. So next, let's start with a wonderful panel discussion. And for this, I would like to invite our first. I'd like to invite both the moderators first. Just a minute. Sorry. So I invite Gaurav Khan sir. Sir is senior embryologist at Akanksha IVF Center. He's the first Indian embryologist to be certified by both prestigious international societies, that is ASRM in 2016 and ESHRE in 2017 and 16. Sir is also certified by the IFS and Global Fertility Academy, have 11 international presentations all over ASRM, ESHRE and IFSS. Welcome, sir. I invite the next moderator, that is Nishant sir. Sir has done his MSc in biochemistry, MSc clinical embryology from University of Leeds, UK. He is currently the scientific director and chief embryologist at Vasundhara Fertility Center, Nagpur. Is also the lab director at Icon Hospital and Nucleus IVF Center, Nagpur. Sir is also the chief embryologist at Medigrace University, Verma TTBSc. He is practicing embryology for more than 2 decades. Welcome, sir.
Thank you, Nidhi, for that wonderful kind words.
So I would request you to please introduce our esteemed panelist.
Right. I think this is going to be an amazing panel because we are having absolutely the OGs of embryology here. So our first panelist that I would like to welcome, it's been my honor. It's Dr. Suresh Katera. I think he does not require any introduction. He's the scientific director of Pearl Singapore Fertility Center, Chennai, and director of Asia Pacific Advanced Fertility Center, Bangalore. He's got a lot of accolades, and everybody knows about him, what he has done, and he's been a teacher to lots of us, and I'm very happy that sir is here for the panel, and I hope we are having a great enlightening discussion on sperm structure and morphology today. Can I have the next slide, please? Yes, so we are having Dr. Mahendra Bhalodia. He's the lab director of Babeshi Fertility Institute, Ahmedabad, Mumbai, Delhi, Vadodara, and Surat, Bhuj. He has done Master of Vet in Sciences, also since 2000 in embryology field, and 23 years of embryology experience, more than 25,000 plus ETs and 2.5 lakh oocyte ICSIs and 2,500 embryo biopsies. So I welcome you here again. May I have the next? Yes, obviously, no introduction at all. Swami sir is there, and I think it's going to be amazing. So welcome, sir. Great to have you with us. Wonderful. We are having Durai, who's also a chief embryologist at Krishna Institute of Medical Sciences, Hyderabad since 2009. He has more than 17 years of experience and has visited West Africa as consultant embryologist, serving as executive member of Academy of, that is our ACE, our body, and has received Best Embryologist Award for Mercedes-Benz Health Summit 2019. Welcome, Durai. Great to have you here. We are also having Hetal Kumar R. Shukla. He is from Nova Fertility. He's the chief embryologist. Great to have an embryologist with us. We are going to have a great conversation here because embryology, fine, but embryology is amazing. So he's got the Icon of the Year Embryology National ET Health Award. Also, he's member of ACE and ESAR, visiting faculty in Gujarat Biotechnology Research Center, Gandhinagar. And he's done a lot of papers, chapters he's written, and welcome you. So let us go to the next one. Obviously, Sanjay is there with us. Great to have you with us. He's a senior consultant embryologist from Pune. He's done his MSc Biotechnology from YCM University. 14 years of rich experience in the field of embryology. Got trained for PGT biopsy from Dr. Alpesh Doshi at Aspire, Mysore. Hands-on training in origin tissue cryopreservation organized by Asia Society of Fertility Preservation, Delhi. Has authored a chapter in textbooks and active member of National Fertility Organization like AACSR, IFS. So I welcome you, Sanjay. So this is an amazing panel that we are having. But where is our co-moderator actually? Oh yeah, Gaurav.
Hi, Nishad.
So first of all, I welcome Gaurav, my co-moderator, and over to you. Please go ahead.
Well, can you see my slides here?
Yes, sir, it's visible.
Yes. Am I audible?
Yes, sir, very clearly.
Thank you so much. Thank you so much, Ahera, Dr. Ved Prakash, Dr. Sanjay, Dr. Charu, for inviting us for this amazing panel discussion on sperm morphology and how we can manage for the best outcome from poor sperm. So, see, I'm a lab man. We have the experts with us, yes. So I'm a lab man. My concern with the sperm is, by looking at the sperm, can we tell whether it has an intact DNA? Can we tell whether this has the exact genetic component what we want? And can we tell whether this particular sperm is apoptotic or non-apoptotic? Just looking at the morphology. There are many questions to see. It is impossible, I know. So how we started, first of all, that what is this normal sperm, how it looks. So it started there was no ray of light which comes and tell us, yes, this is the normal sperm we all should use. Rather, there is a work of lots of scientists behind. It was 1966 by Freund. He was the first one. And what he did, it was very simple. He took all the fertile man, take the sample from them, and on the basis of that, he said, this is a normal sperm, because all normal fertile male, they have this kind of sperm. He labeled sperm like that. Later on, we went into the PCT and the sperm, those are attached to the zona pellucida, and we came to this particular sperm, which nowadays we call it normal sperm, by Allison in 1971, right? So for me, not looking at the morphology, but at the outcome, how I can say it is normal or abnormal, I will look at the spontaneous conception rate. I call it here as fertility. The difference in the IUI results, the difference in the IVF outcome, and the difference in the ICSI outcome. On the basis of that, I can say whether this sperm is normal or abnormal. So I'm going ahead with my questions with my esteemed panelists. Consider me as a newbie in the lab. I have lots of questions with you. The case one is a female. She is 29 years age. Male has 30. 2 years of marriage. Primary infertility. No female factor at all. Male sperm is 2%. Rest all parameters are okay though. So what you will do? Anyone? Dr. Swami, the patient comes to you with this report. What you'll do next?
What, 2%?
Yeah, 2%.
Normal.
2%
Yeah.
normal.
Yes. Yeah, in 2% we are doing the normal ICSI, nothing to do.
Yeah. If it is no female factor, 29 years old, why can't we do IUI?
IUI should be the choice.
But don't you want to check from the other lab? Or we just want to say that somebody has said 2%, it is going to be a 2%?
Yeah, obviously we have to do analysis. So we probably by the
go ahead.
Yeah, we have to do analysis. Obviously by the probably I prefer to the embryologist, the person who is performing the sperm preparation. They should do at least 2 times they have to do in a given center, probably by the same person. So then you decide, and if no female factor, probably I can go with IUI one cycle. 29 years old, no problem.
We'll go with that. So how is experience with this lab variation? Because my point of taking this sperm morphology so seriously, I want to know how much people know about sperm morphology. Because
obviously now, now we started speaking more on morphology. Yeah, others can comment.
Dr. Hetal.
Actually speaking, the real question is morphology being assessed correctly? That is the larger point every lab has to focus. Because, with the WHO cutoff value of 4% to call it normal, and another recommendation from the WHO that borderline should be considered as abnormal, and with that, and many a times, the embryology team also many a times they have made assessment of sperm morphology as a defense mechanism to the fert fail or any other things. So with this, I would say that this should be given a very prime importance for the correct assessment of the morphology, and with that point, a calibration of calling a normal sperm should be made very prime importance.
Any other comments from the other
Yeah,
panelists?
yeah.
I think what
Yeah, I would like to share, this is the semen analysis is the only parameters that we could judge the malefactors. So we have to do the proper semen analysis, every aspect, every parameters, not including the morphology, but every parameter we should value very well, because this is the only parameters analysis for the malefactor. Otherwise, so we have to do very perfectly.
Yeah. Suresh, you were saying something.
Yeah, I think you're asking whether there is a inter-person variability or variability, and what kind of stains they are using. Again, depends on that. Your morphology percentage may vary depending on that. Osmolarity is different. It may distort the morphology and different forms, positive or negative kind of information. So you have to do it in your own laboratory repeatedly. It has come from laboratory. And then make a decision whether you want to go ahead with the IUI or
We'll come to that. We'll come. The main purpose of for me to talk about the single report, and you cannot rely on it when it is coming from somewhere else. You need to check it at least 2 or 3 times before quoting someone as teratozoospermic. So even there are many studies, but this is a French study which says there is more than 42% variation in this sperm morphology than other parameters when it is seen by different embryologists. So you actually have to have a very trained embryologist to quote or label sperm semen sample as teratozoospermic. You cannot take it very lightly.
I just like to add one more question to this is, like we are talking about the staining, right? Dr. Suresh, you just mentioned. So nowadays we also have those, pre-made slides that we are using. So would you recommend to use these slides? Because morphology assessment, if you do it manually with different dyes, it takes a lot of time, almost 2-3 hours completely, So would you recommend using those slides, like for example, the Easy Morph slide or any other company slides compared to doing it manually in today's era?
Yeah, I mean, it might be easier with that.
Yeah.
But then we have to also consider the cost factor in this situation.
Right.
If you look at the staining, I mean, it's a perpendicular staining, which is the gold standard. Now, of course, they have certificate as well as test templates. So it depends on like whom we want to use it, depending on the patients.
Right, because the time frame is a lot. Like in 10 minutes, you can start with the morphology, or you have to do at least 1.5 hours, 2 hours. So that is important if we are a very busy kind of a lab. Right. Okay.
Even I think Nishad, I think always in my center basically, I am using not using that kind of pre-stained slide. I am prepared to do the morphology by myself or my junior embryologist itself by staining, not by pre-stained slide. Never. I never used. I tried 2-3 times, but I was not satisfied with that slide. So I am using that my own stain for that doing the morphology. I think it's always better. I don't know. I don't know. Actually, when I have seen that slides, I'm not happy with that kind of stain of that because maybe sometime it is not show up that proper visualization of that morphology of sperm.
Yeah.
So that's why I'm using that dipquick or sometime pepinoculus stain.
Yeah. Okay.
Yeah.
Right. Okay.
Maybe Dr. Hetal is using. I think he can more elaborate on that.
Yeah, I think so. Yeah, maybe. What's your view, Dr. Hetal, about this?
Actually speaking, pristine slides have some problem of visualization because
Okay.
of, the point I would like to make here is we are putting a drop of semen and then putting a cover slip on it, and
Right.
we just wait for few minutes. So it sometimes you will be seeing clumpings into it, and you may not find spermatozoa in a plane where you really want to see them. So their plane display is not as perfect as, and Dr. Ved is
Yeah.
saying correctly. But when we face this problem, we have changed preparation of this slide on the pre-stain slide by not by placing the drop and placing the cover slip, but same like smearing.
Yeah, exactly.
Smearing,
You have to
and
drag
that
it.
has made significantly acceptable view on the pre-stain slides, and that makes a very useful uh, tool to
I
use
think
it.
it's a very important point that you have mentioned. Even that is what I do. I've shifted a long time ago into pre-stain. And yes, if you just put it, it makes a lot of difference. It will not give you what, as you mentioned, but if you smear it, yes, it's almost
Correct.
close to different. So it does save time, but as Dr. Suresh mentioned, the cost factor comes. But I have realized that the cost is not really that much if you go for morphology, as it saves a lot of time. But yes, it does make a difference a bit. So yeah, let us move on to the next question then.
All right. Last point I want to make here on this.
Yeah, please.
Because as we have seen in the report, one said 8%, second said 3%. Even the WHO 2021 says to find out the difference between 3% and 5%, you have to count 1,500 sperm by a highly trained eye. Morphology is that much difficult to actually observe.
Anybody is doing that?
No, we are counting
I'll talk,
200.
I'll
Yeah.
talk my own intention about the sperm morphology later in the my panel. What I feel about sperm morphology. Although, do you guys doing this, counting 1,500 sperms to reconfirm between 3% and 5%? Does it really matters?
Like, actually, we are confirming. We are when we find the more number of abnormality, then we confirm with other embryology, other andrologists also. But at least we should count 200 as per WHO. We are not count 1,500 as per sperm.
Right.
Yeah.
Now I think we can go into the next question. That will be the thing we can carry on with this, the same thing. So sperm manual versus CASA. So I think Gaurav nicely done that. Here maybe we can say 1,500 sperms can be counted with CASA with different machines. Obviously, that depends on what machine you have. What is your take? I think I'll start with Sanjay here. Would you prefer manual or would you prefer CASA? And which kind of CASA, if you have any specific answer to that?
Actually, frankly speaking, I've not much worked on the CASA system, at least regarding morphology thing. But whatever we got introduced and this thing, see, actual technical work is the same. Like you have to site preparation or thing or the sperm selection, then you have to give the commands that this is the sperm you have to analyze and all. The things will be same. Generally, to avoid the error, like we said, embryologist to embryologist. So that will be more helpful. If you compare with the cost, it is definitely not that efficient.
Okay.
Anyone else here?
No, in my point of view, when we compare to manual and CASA, in manual is more subjective. When you are coming to the morphology, it's highly subjective one. But in the CASA, you may avoid it. But according to WHO, it's a CASA is a different kind of issues it's having. It consider it's another one also. It's a morphologically count. It's a consider as a sperm. You say some pus cells or RBs or something is related to the sperm. It will consider as the sperm. That what WHO also it's not recommending CASA for the morphology assessment. It's clearly is mentioned in the 2020 manual.
Right. So let me make it slightly controversial here. So that is CASA, but now AI is into picture. There are machines available, and they do it on the basis of generative learning and machine learning, taking pictures, studying millions of pictures, and then comparing it and then giving the results. So anybody with that experience of using, though I don't need to mention companies here, but AI assisted CASAs, And what is your take, and would you recommend that in your lab? I will start it again with Dr. Suresh.
No, I don't have a CASA as I cannot afford the one. And, I don't think it will add value, in a routine, assessment of morphology. Again, I mean, I would say, like, recover what we have invested. We need to, pass on the cost to the patient. And how it will benefit. I mean, practically, if you look at it, when we do a morphology assessment, we're looking at either, IUI or conventional IB.
Yeah.
Whereas when it comes to, ICSI, how does, all these, additional, things will help? It is you actually as an embryologist, and what you think is normal, in a vector preparation. You pick those sperms actually, all these, additional things that you're doing, I don't think it will help you practically, while you're doing ICSI. I think
All right.
Actually, sorry. That is the issue. There are no non-invasive technique to find the good sperm, which one actually I used in ART technique. Yeah.
I think we'll go to that part later. I mean,
Yeah,
let us
yeah.
concentrate on morphology. Let us not go into the genotypic part, that will come later. Only the phenotypic, like normal sperm versus abnormal sperm. That is what we are trying to go through. This question is whether AI-assisted CASA is better or at least equal to what indiscrepancy we have with every participant who is doing the semen analysis for morphology. Does anybody has any experience here?
So, may I speak?
Yes, Saili, why not? Please go ahead.
Yeah, so I have some experience on this.
Yeah.
Because we had advised one company for their AI-based semen analysis, and the idea was to try to compare it with manual semen analysis. The problem that we saw in one of the laboratories that we were using it in is it works well in 80% of the cases. We saw for moderate samples, I mean the samples that are in between, in terms of count, somewhere between 10 million to 40-50 million, and somewhere morphology is also, above 4%, or something like a normal zoospermic sample on the lower quadrant.
Yeah.
On that quartile, CASA and manual good is doing well. But when it
Yeah.
is very high or very low, there is a huge difference. Like
I think I totally agree with you here.
yeah. So and we also had debris issues.
Yeah.
But that could be a machine to machine variation. But we have worked on two machines, and both of them cannot detect debris different from semen. I mean the sperm count, and that causes some problems. So
Okay.
yeah, that was my exposure to that. Yeah.
Right. Wonderful. Anybody else?
No,
Probably experienced good embryologist won't prefer CASA or AI. He will do himself. That's all. Simple.
Yeah, the
that's
problem I think is it is used so much in tier 2
Yeah.
cities. That's where it has found its niche. The sales niche is tier 2 cities where there are not good enough number of embryologists or technologists that are well versed in it. But the problem is they are also not well versed in preparing the basic for even for CASA. So that's a gap. That's a gap that can be filled by knowledge and competency. So that's it. That's what I wanted to say.
Who feeding the AI also important.
Yeah, very true. That is another. That's very true. Now the problem
AI
is
itself is a question, sir, but let's go in. That's a different question.
Yeah, we can
That's
have a
what
debate
I
about
don't.
that or a
That's what
discussion
I don't.
maybe. It's a huge one.
Yeah, that's what I don't talk much about AI.
Yeah.
It's a question. Yeah.
Right. So
In the AI, basically what we are discussing about, it take the pictures. It have thousands or 2,000 pictures. The size which is taking is, we don't know. Maybe the focusing, we have the different field it will focus, it won't. It will the same focus in the different, if you are the manually when you are doing, you will adjust the space and you can check more field in the slide. But in the AI, it will focus, it's a fixed focus. It can't rotate, right? That what the techniques, what I read, because we want to some in-depth study on it. Then we come to know that is a fixed microscope. It is saying that it focus, take the 200 picture in the fixed field. How it can be right? Then one more observation, what we observed, it can't repeat the same result, the same sample. If you are first time you are keeping the same and it is giving one report, the second time it is not repeating the same for the same sample. So every time it is started giving the different because of the which the field is taking the picture, this report are keep on changing. This is the I think this is the main drawback when you are using the AI based. The AI means is analyze the image. It not direct CASA, it's directly analyze the velocity and something. But in the AI based machines, it analyze the picture. First it will take the picture, then analyze, then the field of the picture. This will make a big setback to the AI based machines.
Okay, fine.
As of the day, hello.
Yeah, go ahead.
As of the day, I have no knowledge that whether CASA is able to distinguish globozoospermia or not. And so, completely relying on CASA for morphological assessment may lead to some.
Thank you so much, my esteemed
So
panelists.
because of that only, because of the 1980 CASA, first CASA was introduced. After 43 years also, still it is not in the secure the proper place in ART field. That is the
Thank
reason.
you so much. Thank you so much.
But CASA can be a good tool to train and calibrate the people for normal spermatozoa.
I'd like to add something. So I'm doing AI, I'm doing CASA, I'm doing manual, and I've got 4 people doing it. So one report is done by 2 machines and 4 people. And we are fine. Yeah, I mean, there are different CASA. Sometimes your normal CASA gives you readings about asthenospermia or motility fine. And in AI, it is bad. But the morphology reading is matching with our team as well as with the AI. So there are some indiscrepancy. Maybe the motility matches with manual. Sometimes the manual matches with the CASA. Sometimes both matches. So there are definitely problems. The thing is that we need to give more time. And right now we need to do all of them and then start believing in the machines. We should not completely give up on these machines, but we should not complete trust also.
What about count, Nishad? Count?
Maybe I'm discussing morphology.
Just for addition.
We have a whole panel. Thank you so much. I have created the controversy in the initial part only. My main objective of putting these questions is we cannot rely human for sperm morphology and we cannot rely on computer also. So sperm morphology is actually a very controversial topic. You actually need very trained eye to say this is abnormal. I remember the Daniel's remark on me when I said, what is abnormal, what is normal? He said, you put a picture of the sperm in your against your bed, and when you open your eyes every day and see whether it is normal or abnormal. This much of consistency and hard work you need to say this is normal or abnormal. Thank you so much. So now we are going ahead with the same case, and you checked yourself now. This sperm morphology is 3%. You are sure, you are trained by WHO, you know your work, you have good experience of more than 10 to 15 years. Now it's 3%. Will you give chance to spontaneous conception? Do you think these kind of sperm can conceive, with this kind of sperm, a couple can conceive? They can have baby with 3% or even 0% of sperm morphology, or we should directly take them to IVF or IUI? Because this is a normal question. A patient come to the OPD with this kind of report and say, what I should do now? The main question arises here. What we should do now? Should we ask them to try again natural, IUI or IVF? Anyone?
Definitely they can go natural.
Yeah.
Yes. Already we
Count is not a problem. Yes.
Now 3 to 5% of WHO itself telling 3 to 5% you can count more, but it considers a normal, right? So definitely they can try a chance with natural conception. There is no doubt about it.
Sanjay, you want to comment about absolute teratozoospermia? I heard your discussion regarding it.
The paper also.
I think my paper was
Yeah, I was just searching, so I got that paper. I think later you can tell it detail. See, for this 3% thing, I think we should actually look after exactly what type of other sperms are abnormality having. Like suppose if it more the percentage about head defect or globospermia, to better you can go for ART technique. If it is the neck defect or a cytoplasmic ring, then we can think of IU, I think that way.
So you don't want to give them a chance to try naturally again for 6 months. You don't want to observe them under your control.
See, if that way, otherwise, if female factors are normal, including the tubal patency, the better you start with the ART treatment. Otherwise, they already tried for a year, then that is the thing, yeah.
I want to tell the audience basically that 0% is not the indication for the ART. If you
3%.
0% normal sperm, you have all teratospermic, it's not an indication to go for ART. You can try it naturally also. Though you are right, if you have some specific monomorphic abnormalities, you can go ahead with the ART. We'll discuss that later.
Agree.
In the same paper, 24 patients were taken. All 24 conceived naturally in the second attempt. Even those who conceived naturally with 4% morphology, some of them needed ART later on. Very beautiful paper. So what about IOI with 0% sperm morphology? There's a beautiful meta-analysis. People are sometimes worried. I've seen people coming to me and say that we have poor morphology of less than 4%. We should directly move to the ICSI. This is the right way of doing it. We should not spend more time because it's not going to conceive. They have a very poor chance of conception with the poor sperm morphology. And this is a very beautiful meta-analysis, the best form of paper which is available to talk about IOI. And they have gone ahead with more than 4%, less than 4%, less than 4%, or more than 1%. Less than 1%, more than 1% sperm morphology. And the outcome was similar. So if you have sperm morphology of less than 4%, more than 4%, less than 1%, or more than 1%, the outcome, the pregnancy rate and LBR with this meta-analysis says, huge meta-analysis is that no difference. You can go ahead with the IUI. Nobody's going to stop it for this. Now, I want to ask you about IVF or ICSI. Poor sperm morphology is there. What is your option? Do you go with IVF or ICSI? You want to try IVF with the absolute teratospermia or poor sperm morphology. Swami sir is here. I saw him coming.
Yeah, I'm here only.
Yes, sir, please tell us.
Yeah, if it is this scenario, I may try once IVF.
Okay. You want to try IVF? Any other comments?
Because I think nature is better than me.
No,
in 2 or 3% abnormal form, normal form. So we are also offering the IVF to the patients.
If you have good number of oocytes, go for a 50-50. That would I would say.
50-50 also.
Yeah. If less than 5 or less number, go for ICSI. That will be my choice.
Yeah, if you are following the double layer density gradient preparation, it may reduce what the literatures are showing. It has the better normal sperms. So 3% anyway we are planning. If you are using the proper preparation, it will give the better. You can go with IVF. If everything in another parameters also agree.
I think I want to add one thing, because we are not considering here that other parameters. If volume is good, concentration is good, why to go for ICSI? We can directly go for IVF, or maybe IUI you can try. Or age there is no age factor, then definitely IUI can work for that patient. I think so. This is my opinion.
Sir, the point of putting these words here is, I want to tell my young people, those who are coming to the field, that if you see sperm with poor morphology, directly don't jump for ICSI.
Yeah.
There are enough evidence which are available.
Yes, you are absolutely right. Yes.
In absolute teratospermia, you do IVF, and there is no difference in the clinical pregnancy rate, fertilization rate, live birth rate, whether you're doing IVF or ICSI.
I would like to add something here.
Please.
Even like in patients, when the morphology is above the cutoff, we have seen actually fertilization fail depending on how you score, about roughly 10% of population. Now, if you have to take into consideration the motility as well, the forward progressive motility. The morphology may be 3%. If the progression is poor, then you cannot try any other method other than it's that's a way forward rather than actually trying things.
Can I go ahead?
Yeah,
Thank you, Tom. Thank you so much, sir. So even in the sibling oocytes, people, they have tried in different patients, but even in the sibling oocyte, they have tried and they found no difference in the outcome. They went ahead with different forms, even the sibling oocytes, in isolated teratospermia, more than 4%, 3%, 2%, 1%. No difference in anything. So we guys are more considerate about sperm morphology at all together. But the outcome, what the data says, what the study says, is absolutely in contrast. And they say that no difference at all. To save the skin, I know we all are worried about saving our skin and saving our patient. We do ICSI. That you can do in 50-50. But if you're confident with your IVF results and outcome, you can definitely go ahead with the IVF if you're going with the IVF in absolute teratospermia. Now, case 1, we are going ahead. You take the chance, you checked it, it's 3%. Patient conceived, but aborted at 3rd week. The same patient conceived and aborted at 3rd week. So do you think that this is going to give us some link between this sperm morphology and the recurrent pregnancy loss or pregnancy losses? Do you think somewhere it is related, sperm morphology with pregnancy losses? Dr. Bhadolia, Hetal, anyone? Because these are the doubts which gonna strike us later. That patient conceived, we did the ICSI, patient conceived, come back to me. 3rd week, she aborted. So I'm gonna look at the female side, I'm gonna work on the male side. What should be my concern now? Anyone from the
we have to look at the quality of the sperm, not just in addition to phase. I mean,
little loud, sir. We cannot hear you. Little loud, sir. Please.
Probably, we are looking at the sperm DNA fragmentation. Probably, that could have contributed to this. So, I mean, there is probably a divided opinion actually.
Right.
In semen analysis, sperm DNA fragmentation test for all the patients to ensure that whatever sperm you change it, at least normal DNA composition. So I think that has to be looked into rather than doing a trial and error like this. It should have been done in the first place semen analysis.
Right. So I think that's a very good take-home message that that phenotypically good-looking, normal-looking sperm does not mean it is genotypically normal. So morphology is something that we have to be very tread lightly. Although we've got a very nice 3% thing, but yet there is a miscarriage. So you have to be careful with that. Yeah.
Okay. Can we move to the second question now?
Yeah.
Okay, the case second is here. Female age 35, male 45, 10 years of marriage, secondary infertility, एक हंसता खेलता बच्चा है घर पे, no female factor involved, male sperm morphology 1%. They are trying and not able to conceive from last 2 years.
So in consideration of the female age and 1% morphology, we should go for the ICSI in that case. Because in 35 years, there is a less chance of natural conceptions also.
Okay, and any other panelists want to see this and comment something else here?
I think first thing first, the clinician angle pathology has to find out exactly what can be the reason, considering that he already has one child and maybe later there are some changes in his sperm parameters. So he might prescribe some antioxidant and all, then give a try. Then we can take a decision. See if there are changes in sperm parameters. It is lifestyles and all. Yeah.
Okay. Anyone else? Swami sir?
I will refer to the next clinic. No, actually,
We'll refer to the next clinic.
Actually, see, maybe probably as Sanjay said, see, the problem is that past 10 years, what happened to the sperms that we have to see? Like, not much change is genetical. Like, our lifestyle changed. That's because of this problem is, persisting, and we always deal with poor sperm quality. Like when they say normal sperm is in 4% even in the normal male population, and when they come to us with substandard quality in gross border level, our task is much bigger. The entire the episode of morphology looks very simple, but as and when you do experienced embryologists, you select the sperm. That's why most of our ICSIs are normal. So that is how the experience. See, if you ask me how I'm defined the normal sperm, I don't know, but I select the normal sperm. If you ask me how I'm selecting the embryo, normal embryo, I don't know, but I select. So maybe probably something which is past 30 years, 25 years of experience, we are doing that. And remember that when we deal about sperm morphology, it is always a risky thing. And beyond that genetical level, there is a small correction mechanism which oocytes is taking care. That's why we are all surviving. Thank you.
It's your personal brain and neural network AI working to select this embryo.
Yeah.
So I want to bring your notice to this particular study which says that there is a decline in sperm morphology. I'm so happy with you guys that you guys have not considered sperm morphology here. You said it exactly, there is no problem. We know that sperm morphology is going to decline with the age. But we all have seen that female age is 35. I'm so happy with you guys that you are considering male, that they're refreshing their sperm every day. They're making their new sperm every day and making it for a normal conception. Thank you so much. Cytoplasmic droplets. Now we are going to the monomorphic forms. How many of you are able to differentiate the cytoplasmic droplets in this sperm? Is it really a concern when you are doing ICSI or IUI?
I don't think.
Not much.
Dr. Suresh, I want to bring your notice here, please comment.
I think, we try to avoid it with sperms and cytoplasmic droplet, but I understand, it depends on the amount. But there is a large amount it can generate to ROS. It can be damaging for the oocytes, probably. And then sometimes, when you pass through your exhibit, it can actually sometimes come off actually. So you could do that a little bit of manipulation.
Okay. Anyone else? Dr. Hetal, Durai? Have you seen sample?
I'll just speak to this.
Yeah.
See, first of all, cytoplasmic droplet means that is the spermiogenesis has not been completed, complete maturation has not happened. So from histone to protamine 1 and protamine 2. So this is where the change of your genome takes place, covering. So that is another reason where the oxidative stress is there or not, it is affecting. That can also affect and can lead to fertilization failures, or it can lead to further problems on DG. That is your late paternal effect, where the blastocyst formation or the quality of blastocyst, maybe a C-grade blastocyst might be obtained in such a case. So cytoplasmic droplet is a thing that you should be worried about and you should be careful while doing ICSIs.
So now we are coming to the monomorphic form, and I also bring to notice this. Then when you have excessive residual cytoplasm, so there is a difference between cytoplasmic droplet and excessive residual cytoplasm. When it is there, when you have around half of the sperm's head, you have the droplets there, then it's a concern. You may have more immature sperm. And when you go with IUI and other forms with this, you may have a poor results. Definitely, ICSI is a better choice because you have a tool to select the sperm. That's ICSI. And your eyes, definitely. Now, what we can do in the lab with the monomorphic sperm abnormalities, we have talked about globozoospermia. So somebody was saying that whether we can see globozoospermia or not. I saw Sanket talking about partial and complete globozoospermia. Can any one of you differentiate between partial and complete globozoospermia? Any one of you? I cannot. I'm telling you, I cannot differentiate.
I think it depends on the shape of the sperm head. You do, occasionally, very, very rarely, you see, sperms with a little bit, protruding a bit of, acrosome actually. So probably that's it. Otherwise, it will be like, dumbbell shaped, totally, wrong. And that's probably 100%. Sperm.
Sanjay, can you differentiate between partial and complete globulospermia? Can you see the globulospermia, how it looks?
Yes.
Sanjay? Gone. Durai, are you there?
Yeah, I'm here, but
Yeah, please tell me. Can you differentiate between the globulospermia? Can you differentiate the globulospermic sample? How it looks? Basically, you have to tell a young newbies, those who are listening to you right now, that how a globulospermia looks. Whether we can differentiate between partial and complete.
That's what I'll explain what I seen in the microscope, how to convey now here in theoretically.
Definitely, we want it. We want to experience that.
No, actually, when the globospermia, it's not completely like globo means round. We refer as a round shape. Sometimes you are able to see in like in the picture also you see in the stains here. You see that except the spherical shape is missing. But it looks like I feel it's that level only. What I understand myself in the globospermia. But the shape of the head, the spherical shape is missing. It look like normal flattened one. What I see that I consider as a globospermia, then I will proceed. That kind of mostly if it is like Sanjay said, partial or fully globospermia, I'm okay to do ahead. Otherwise, if it is 70% also, I feel is a globospermia. Declare is a globospermia in my safe side. And what are the alternate, I will look it for it.
Completely agree with you. To differentiate between the partial and complete globulospermia, we need electron microscope.
Okay.
In our labs. So whatever you see as a globospermia. What you get, what you do with the globospermia? Do you go with spontaneous conception, IUI, IVF, ICSI? And if ICSI, how? What do you use for this with ICSI?
In that case, if you are asking me, I can go with the ICSI only because anyway they are already infertile. They are already spent enough time to consider this all will not happen. So I directly will go ahead with the ICSI. That is the least option because I don't want to prolong the time because maybe that could be the reason they came here to land on us.
So we all are coming to the same conclusion that another monomorphic abnormality, except after ERC, excessive residual of cytoplasm, we have globospermia. Whenever you see a globospermia, you go ahead with directly ICSI. Now, do you go with ICSI only or you artificially activate the oocyte?
No, first, if it is the first cycle, I can't go with 100% oocyte activation because sometimes if it is having the chance of more oocyte, I can 3-4, I'll go with 70%. I try with only ICSI without activation, oocyte activation. If it is possible, I go with 30% with activated oocytes.
Anyone else? About you have 10 oocytes. Globospermia, what will do?
Activation. I think for all it's clear because as such, the fertilization rate is low, and I think activation must be.
I agree. So anyone for the public, because I got lots of messages because I put a video on Instagram about oocyte activation. I got lots of messages how we do oocyte activation. करते कैसे हैं इसे? People don't know it actually that what we use and how we do it. Any one of you doing the oocyte activation, please for the audience tell them so that they get some message from the discussion. Yeah.
After it's you expose in this.
Yeah, we have calcium minus for
10 to 15 minutes, and then you have to wash it several times, and to get in your culture properly.
Okay.
It's not just we'll export to the calcium ion high concentration of media. Yeah. And as Suresh has said, we can just 10-15 minutes, then we can wash it. That's the important. We have to wash and keep in the culture.
Very nice.
It won't make sense.
So we have to warm the media basically because it is HAPIS based. These are calcium ionophores. Their work is basically to increase the calcium import inside.
Concentrations, yes.
So we have to put after ICSI, performing ICSI, you put the oocyte in the media for 10 to 15 minutes and then wash thoroughly 4 to 5 times. And then you culture them. And the next day, what is important is to look for 3PNs because these oocytes after calcium ionophore activation may have parthenogenesis. There are incidences of 5 to 10% of 3PNs. So you have to be very careful to exclude or deselect all 3PNs from the fertilization set.
I would like to add one more thing in oocyte activation. Do you remember that manual method while you're doing ICSI, that you have taken the cytoplasm and swirl the cytoplasm? I think it was shown in ACE Goa or ACE Chennai.
Goa.
Yeah, Goa, right? And I remember I had a very good discussion with Priya, and actually we did that and we did get oocyte activation. So it does work. Obviously, it's an adjunct, but just like calcium ionophore, it's also an adjunct method. You can try this also if you're good at XC and still get your oocyte activated.
We can only worry about the damage of the oocytes. That's it.
Yeah. So you have to be very careful. It was some guy from US who had done this. He's published a paper on that. If I remember, I'll send it to you.
Sure.
There are a beautiful comparison between the mechanical versus chemical oocyte activation, and with mechanical in global zoospermia you have 30% fertilization rate, and with chemical you have 70. So it's better you go with chemical. And as Durai said, you poke the needle and you do this, there are higher chances you're going to make a bigger hole and everything going to come out. So choose the better way, which is actually rational also, right? Macrozoospermia. Another abnormality I want to talk about. These are the patients where we need consideration. वो 3% 2% तो conceive हो जाते हैं. इनका क्या करना है? Lots of flagella, oat sample. We see 2, 4, 10 sperm, and they all are like that. Can you do something with these kind of sperm? Anyone?
Have higher, incidence of aneuploidy. So such sperms, when you do that, probably you'll have to do a PCT and whatever.
Durai, have you ever come across this kind of sample?
No, I've seen double head, but not this multiple head. In my experience, sorry, I didn't.
So we have not come up with the 100% macrozoospermia. We have 4, 5%, 10% macrozoospermia. We have chance to select the normal sperm of that also. Like global zoospermia, we have not seen the 100% macrozoospermia.
Bhadoria, Swami sir, please come in. You want to say something?
No, my question is that, we have all suboptimal sperm, morphologically not good. Oocyte we have to activate. Everything adjuvant will give and expect the normal thing. Full term. So this is the problem. See, there is a percentage, 1% we are seeing like these are patients. And we have to remember one thing, we will not see all abnormality in one sperm.
Yeah.
Understand?
Yeah.
So that is where we have given the freedom to select the sperm, embryologist. Oocyte we can't select. Clinician will give. Sperm, we have given the freedom to select. So the people who are all coming to us, not everybody is, millionaire. They may have less sperms and abnormal sperms, morphologically, compromised sperms. So one should develop that skills, what I say, to select the best out of that. That is my message, that's all.
So this macrozoospermia, they have another issue. They have a big head. The head size is around, they're diploid. They have around 6 to 7 micrometer size head. So this happened with me actually. Yeah, please go ahead, Dr. Swami wants to say something.
No, you carry on. I am listening. Yeah.
So what happened with me, I chose a sperm, and when I was doing ICSI, I was picking the sperm and the sperm was not going inside. So we have a very simple machine with us to select the sperm, that is the ICSI needle. This bore or the inner diameter of the ICSI needle is just 4.5 micrometer. It will not allow you to pick sperm which is actually having a bigger size. Be very careful when you're picking the sperm. Don't push it, because they're actually a cell. They can squeeze and go inside. So whenever you're picking the sperm, be very careful whether it is going easily inside or not. If it is going easily inside, you can select the sperm. If it is showing some kind of resistance, avoid that kind of sperm. Any other comments from my panelists? Sanjay, have you ever experienced this kind of sample? No, Sanjay is gone, I think. It's the party time. Okay.
Sorry, bro. I was on a call. Yeah, sorry.
No worries. I have a new question for you. Come here.
Yeah.
Now you see a semen sample with no sperm motility. I'm saying it again, no sperm motility. All are
Yeah.
immotile. What you'll do?
Yeah, obviously host test first. We'll do that.
Okay.
Yeah, for analysis purpose. And then for ICSI, we'll use something like sperm mobile. Yeah.
Right. So what do you want to do with using the horse? What is your idea behind it? What do you want to differentiate here?
Yeah, see, actually, with the uh, potent sperm or live sperm, which is immotile, the cell membrane will be active. So it will say changes when there will be a hydrolysis or water intake, then it will show curved tails. That is the sign of
Yes.
the coiled tail. It will be the sign of a live sperm. Yeah.
Right. So you know what happened to me once? I saw a sample of this no sperm motility. I used horse and 70% were viable, and my clinician, some different clinician, wanted me to do IUI by adding pentoxifylline in it. Can we do that?
Okay, and for IUI, we need a motility. Obviously, it is not possible with that. Yeah.
So, do you think that's a clear indication for going ahead with the IUI? Do you think can we go with the IUI with no sperm motility by using pentoxifylline? They start moving, right? They start twitching a little bit. Can we do IUI?
So IUI is minimum 5 million motile sperm should be needed. Then it is no use of doing IUI.
Reaching
5
sperm cell
million is
and
the total
reach
amount.
the fertilization, that's the question.
Sorry?
Reaching sperm,
yeah.
And when you do IUI, it has to travel to the ampular region. And they reach
Right, I think you just answered the question. And I believe now, like Gaurav mentioned, pentoxifylline has been known to be toxic. I think we have shifted to theophylline. So that is one thing. And with horse also, horse is still relevant, I feel, because even after horse, because it's a vitality test, you know that the DNA is intact, like it is functional, the sperm is alive, and you can transfer that. Even doing horse with culture water, and then you can do ICSI with that, and that can even give you live birth. So that has been cases. So it is also a safe method of doing. And obviously, so the point is, with no sperm motility, you can do horse, use theophylline and pentoxifylline, and get results. That's what you can do. But IUI, as all you guys are mentioning, doesn't make sense.
IUI, we have to take the sample, go inside. You can't inseminate.
These why I'm saying this, sometimes they're new people, they are new clinicians, they actually
Yeah.
don't know what.
Yes.
So
It's really very difficult, especially. Yeah. Especially in the northern side, it's very, very difficult. I know. Yeah, so too much of expectations.
In that sense, Gaurav, I would like to share one thing. Once my clinician told me to put monocyte into the calcium ionophore so that it will become M2.
You teach her IBM, Ray.
No, better you can pour it to, you can give it to the patient, she can drink.
Folks apart. This is a huge debate on M1 and GVs. Okay, nice, very nice. So this is another abnormality of ciliary defects which Sayali and Sanket have taken. We are not going ahead with what they have said, but when you see this, what lab people can do is horse, see whether they are viable or not, to differentiate between the necrozoospermia and asthenozoospermia, basically. So here, when you have viability, basically they are asthenozoospermic, not necrozoospermic. So when you have asthenozoospermic, you do pentoxifylline, theophylline, go ahead with the using it for your oocyte, do ICSI, and make good embryos.
Great.
Coming to Nishad, please go ahead.
So most widely used method of sperm sorting in infertility treatment, which one would you prefer? I mean, it's in front of you. I mean, we got all the facilities. We do density gradient, we do swim up or direct layering, we got microfluidics, we got max, we got PIXI. I mean, what not do we have? And I would like to ask you, which method would you prefer and why? And is it really relevant? I'll start with Dr. Mahendra here.
Yes, actually, this is all this technique we have to use as per the indication actually. So there are certain mats and microfluidics. Actually, we are using the microfluidics when there is a high DNA fragmentation in the sperm. That type of cases we are using for the sperm preparation. We are using the microfluidics. When we are using the PIXI, there is a hyaluronic binding testing. When we are for the ICSI, where that person when there is a higher DA5 percentage in the sperm. So in ICSI we use the PIXI. So we don't have the IMSI.
Okay. Yeah, anyone else, please?
Yeah, there is a non-centrifugation methods, microfluidics. It's only used when you detect in high DNA.
Could you come closer? Your voice is dropping.
Yeah, can't hear.
For Wi-Fi here. I think you have to use it only in where it is indicated as Mahendra said. You cannot use it for all the patients. Non-centrifugation, which is microfluidics. When you detect higher DNA fragmentation, only you in those patients. Otherwise, it is not required for all the patients.
Yes. Right. Yeah. So anybody has any other things to say about this?
See, if you want to focus on morphology, use IMSI. If you want to rule out apoptosis, use MAX. And PIXI, if you are focusing on selecting mature spermatozoa, because hyaluron binding is the means binding will take place with the mature sperm is the science behind. And motility and relation to the DFI, if you believe that, go with microfluidics.
Fine, because the issue is basically everything boils down to one thing, and that is your DFI. If you see this slide also here, it's technically mentioned DFI except for MC. Now the point is that you've got so many new companies also coming. They are bringing small machines and devices that you put in the sample, and you get post-wash DFI. Well, if you look at it, if you do Max, PIXI, like most microfluidics, the new methods or any methods, post-wash your DFI is going to fall anyway. The only thing is there are some problems, especially in Max, where you are doing more centrifugation, or in density gradient also. If you're doing higher centrifugation, higher Gs, the oxidative stress is going to increase, and then your DFI will increase. So point is, you have to reduce your DFI. It's morphology, yes, we are talking about it, but is morphology the only factor? Is it good morphology is going to give you results? We got earlier questions also where we mentioned that bad morphology or literally zero morphology also gives you pregnancies and live births. So I think the point here is that we need to make a good quality, good genetically normal sperm to be selected. And any of these methods do work, but the only thing is that we have to be careful with the fragmentation of the DNA here.
Can I come in?
Yeah, please go ahead.
Sir.
There are two things. One is sperm preparation, another is sperm selection. Probably in the era of ICSI, we have to select the best sperm. So possibly we have to use the techniques like advanced techniques, which is non-centrifugal, where we can reduce the DFI. We have to look for it.
Yeah. So definitely looking, I think lots of papers also in support of microfluidics, but I think there are also problems related to it. Dr. Suresh, I think you must be aware, I think there have been issues of higher miscarriage rates with microfluidics. Although the blastocyst quality is really good, you're getting very good pregnancies, live births are there, but your miscarriage rate is slightly higher compared to most of the methods. So what is your take on that? Why is it happening? Because I am confused.
With microfluidics?
Yeah.
Miscarriage rate is high, he is telling, like some papers reports.
I'm not, I mean, I know a couple of papers where they have shown reduced DNA fragmentation. But it doesn't increase, I mean, I haven't read anywhere, it increases, the miscarriage rate actually. It should actually reduce.
Yeah, but I think some papers, maybe there are papers, I mean, good sample size also, where they have mentioned that the miscarriage rate was found to be in the first trimester especially, was found to be slightly higher as compared to your density gradient method or even normal methods like swim up methods. So that was a bit even I was confused that why is this happening, and there is no explanation to that. So yeah, if you look at it, microfluidics looks like an amazing method, very easy to use, very easy to manage, and definitely good, as Swami sir also mentioned, good sperm selection technique also.
Shukla wants to tell something.
See,
Yeah.
most people discuss is about density gradient, and density gradient increases the DFI. That is the probably I call it hypothesis or myth. Why? Because uh, uh, density gradient or centrifugal force hardly will cause damage to the DNA. What happens actually is, different protocols of density gradients are in practice in different labs. So with higher application of higher G force and for a longer duration of minutes, that makes probably you will see lot of immotile spermatozoa. Lot of means in good number immotile spermatozoa also come into the they go down to the bottom of where pellet is formed. And these immotile spermatozoa, when you assess for density, DFI, that time they will show you the elevated DFI, and that gives this idea that then the DGC increases the DFI. In this case, it may help that if you have a good sample, good concentration, good volume like that, probably it will if you reduce the G force and time given to the density gradient, and it will have a very good acceptable spermatozoa. That's
Right.
what I.
So have you guys done any post-wash DFI and compared it to the pre-wash?
Exactly. Not only post wash only. I have compared. I have compared neat semen before DGC, post DGC.
Yeah, that's what I'm asking.
And before max and post max also. So
Yeah.
all these I have compared, and then
What difference did you find? So what difference did you find? Like, what was the DFI? Can you give us a number that so and so was before and after? How much was the difference of the DFI index?
DFI has come down to even less than 3%, 1% also. Very rare. Data already I have. I have done it.
So I think if you're a good embryologist, good andrologist, if you prepare the sample properly, even
better.
a high DFI sample can be brought about to a very low DFI with the right techniques and proper mindset that you're having. So yes, that will make a lot of difference here. And then even morphology in some methods, does it also make a difference while selection methods are used here?
Pardon? I didn't get.
The like this is the DFI. So in density gradient, even swim up for that example, if you keep it for a longer time, bad morphology sperm also swim up. So you have to be careful with that also.
I don't know how much true it is, but for swim up, somebody has said me that within 15 minutes, if you instead of 45, 60 minutes, some people are putting swim up for 45 or 60 minutes. Yeah. But mostly it is the practice, right? But within 15 minutes, if you take the supernatant of the swim up, it's ideally good, because swim up is always done with good samples, and 10-15 minutes all good motile spermatozoa will be there into the supernatant. And rather, there is a possibility if you keep it longer, spermatozoa may swim down back to the seminal portion. So that can.
I think what I found, if you keep it for a longer time, the bad morphology sperm also come up because they are slow moving. They slowly come up.
Maybe
that does happen. So that way you have to, the timing what you have mentioned is a very good point, that 30 minutes, maybe 45 minutes max is a good time. If you go above 1 hour, definitely, I have observed over 1 hour, 1 hour, 15 minutes, bad morphology sperm will definitely come. So while doing your methods, be careful with your time, be careful with your centrifugation, that your G, and be careful with your spin time. I think these three things you have to be very careful.
I think 20
I
minutes
think,
is enough.
Yeah.
okay, sir, lovely. So 20 minutes, 15 minutes, any other figure? Anybody would like to suggest here?
Same
Spin
20
time?
30. Yeah.
Okay, lovely. So I think
See,
that's a wonderful thing.
yeah. One thing as just one liner, like techniques, like we pay observation patient to sorry, person to person differs. The actual processing technique also differs from person to person. So while conducting this procedure, you have to be very particular. Like after how many time you take sample processing, overall maintaining the temperature of the sample, these things. Yeah.
Like, okay. And now, since we are talking about these methods, cost is a very big factor. Whether it's IUI, whether it's IVF or HC, yes. Now here's the catch. So we are in India. I mean, forget even abroad also, but this is a very important factor. Now there are so many companies coming us to us that our swim up machine is there. I'm from Israel, I'm from US and UK and all. You try our machine, it's 3,500 rupees, and here we are making samples with swim up and density gradient at 300 rupees. So is it really making a difference? Would you prefer these methods? And if so, yes, is it really worth it? Because microfluidics, as mentioned in the slide here, 4,000. You got your, I think, sperm selection method, that is your Hylase method, that is 2,000. Pixie dish. Max is the most expensive one. And obviously, you've got another one. I can't see which one is it.
IMSI.
It's
IMSI.
IMSI. Sorry. Yeah.
15.
So, yeah. What would you prefer, and would you suggest this to your all or our audience? What's your take on the cost relative here? Swami sir, I'll start with you.
See, cost wise, see, I'm not about the cost, see, because see, IVF itself a costly affair. There is no doubt about that. Only thing, whether we are doing the justification, like, you buy that thing, whether you are, see, each technique is for the particular population. We can't individual, you can't make it ready-made. It's medicine is always tailor-made. There are certain, this developed, there are so much of research done behind every technique, whether it is useful or not to our population, we have to decide. And depending, see, having all the technique is one place also not possible. I can't have EMC, all other, everything in one center. There is investment involved. No, I may want to have that, but my boss, he has to invest. That is a different story. So I think we have to be very legitimate to selecting the techniques or providing this kind of advanced methods. We can't generalize it. Still, I think probably my idea, I'm interacting with people, probably 80 to 90% of Indian centers, we are not doing any, I fund of things like, IMSI or these things. I'm sure probably maybe Pixie you can do maybe
I think
2,000
it's only
rupees.
Max and microfluidics and the new
Yes,
swim
yes,
up.
very, very,
Expensive.
yeah.
These are the three new
Yes.
gadgets.
Yes.
I want to address the elephant in the room, especially for the swim up method, those machines are coming. How do you justify the cost for IUI, which is costing you 3,000 rupees and 3,500, where the whole IUI cost in 2-tier and 3-tier cities in 5,000-6,000 rupees total?
Exactly. Yeah.
Yeah, I mean, how do you justify that? People come to you, there's a company, people will come, they'll talk to you, and they say that our DFI is 3%. Well, if you do the sample, prepare it, do DFI with swim up basic also, it's going to give you 3%. Then how do you justify the cost? Yeah, Durai, what would you like to say?
Actually, this is the cost factor. If I am asking as a embryologist, I go for with EMC. Once investment, we can, whenever the case will come, I can follow it up. These are all, it's when we are doing the tier, as you said, tier 1, tier 2, maybe the tier 1 won't be any difficulty. When you go to the small towns, maybe the cost will be the factor. But if you are asking me personally, I'll prefer the EMC one because it's once invested, I can go ahead whenever the case is lifelong, I can use it.
Yeah, they see, especially this kind of machines, what Nishad is telling is that, they are saying that you don't want anybody at your lab. You yourself can do. That is the catchy thing. You don't want anybody expertise. You don't want andrologists. You don't want embryologists. Keep next to your, consultation table. When the patient goes and gives, you do. everything and do the insemination yourself. That is how it works. I don't think in long run it will work. Maybe they catch all the people who are not very much aware of that the nitty-gritty of the infertility.
Yeah. Well said. Well said. Agreed.
Anyone?
Anybody?
Yeah. Suppose if we go as per that every technique mentioned here, if you go with the max, here we see we want to, we don't want to use apoptotic cells like sperms. So we know that we can avoid more apoptosis by using a fresh semen, like there will be less gap between the ejaculation per sample. Okay, that is one thing with the max. Microfluidics, I personally don't feel it is much different than a classical swim up technique that we directly overlay the motile sperm. Yeah. And the third one is IMSI. Durai will tell better. Other than vacuoles, is there any parameter that it is difficult to identify as a morphologically difficult? Yeah. And fourth one, Pixie. As per ICSI say, chromosome doesn't have that much big role into the ICSI. So, and we are already using the density gradient on less gap between the ejaculation. So we're trying to get more closer to the base sperm. That is what I think. Yeah.
Yeah, Durai would like to answer that. What Sanjay is saying. You've crossed the line of controversy here.
IMSI.
Yeah.
No, that's what it's an investment part. It's only once per investment. But whenever I feel that where the morphology is in the concern, so I can go ahead with that. When I feel that repeated failures, usually what I follow my IMSI. Whenever I feel the 4 times, 5 times the patients will repeating, I may try this. Not no one will tell, but I can take one extra mile to give for my own satisfaction. But otherwise, this in the hopeful, I'll go with the microfluidics. Microfluidics is maybe in future, like what the Somi sir said, they can do now the clinician, the tier 1 clinic, they can just do their own table. They can prepare, put the sample, they can collect and do the IUI. The level 1 clinic maybe must benefited with microfluidics and the sample. Remaining one will work much difference, but as per the population, which sample is needed, the technique based on that we can select and process.
All right. Okay. Suresh sir, I'll end this question with you. What is your take on this? Dr. Suresh, are you with us?
I think he's left.
Okay. Okay. Yeah, I think Gaurav, can move on to the next question.
I think we have extended time also. We are about to finish almost. So we are done with this. Somebody asked here that can this sperm morphology impact the perfectoderm of the blastocyst? So I found one Chinese study, and looking at the sperm parameters, the defect in the tail, the piece, and head, they said where the problem could be. If you have a problem in the defect, you may have the issues in the blastocyst. If you have mid-piece defect, you have T5 to T9, that is in the compaction to cleavage stage issues. If you have defect in the tail, you may have issues in the fertilization to T4. But you know what is the amazing thing about this study? They did IVF for all. So that's why it's a Chinese study. So don't fall for this kind of, don't fall for this kind of studies.
How do we conclude this this part only particular for this?
It's a very amazing study. Go for it. It's very amazing. It's all IVF. How they separated it then? Went where? So don't fall for this kind of questions and trauma in your brain. That
you such the Chinese study.
That's why he said Chinese study, no, first itself.
Don't
Expect
fall for
unexpected.
it. That's a very nice question, but for that you need to inject thousands of oocyte, a different abnormality, culture, then you're able to get to this conclusion, which is absolutely irrelevant until unless you find.
You need to inject thousands of abnormal sperms.
If you do the beautiful study, yes.
Until unless you find some best protein markers which are available in different abnormality of this sperm, then you can definitely correlate. So I'm so happy that my team panelists, a team of panelists, are able to give me exact answers what I wanted. That sperm morphology altogether, whether it is 0%, 1%, 2%, 3%, 4%, not gonna change your spontaneous conception rate, IUI, IVF, or ICSI. What matters here is monomorphic abnormalities like globospermia. Mid-piece, extra residual of cytoplasm. There you have to do XC. Tail defect, macrozoospermia, and ciliary defect, which is very simple to assess because they don't have motility. There you have to apply your brain and do XC. Thank you so much, everyone. Thank you so much, Nishad.
Thank you. Hi, Hera, Ved Prakash. Thank you.
Bye. Thank you, everyone.
Thank you, Ahira. Thank you, all the panelists, Gaurav and Ved sir, Charudat sir, and Sanjay sir for the opportunity. We had a great time, and I hope everybody enjoyed this panel here.
Yeah. Happy Christmas, Happy New Year.
Happy Christmas.
Thank you, everyone. Sir, I request Ved sir to please take over for the thank you note.
Thank you, Nidhi, and thank you all. It was a wonderful panel, and I think other than participant, I also learned so many things, new things from you people, and it was wonderful. And definitely next time we are trying to do same kind of morphology thing, maybe oocyte morphology. We are trying to dissect each and everything in this. And with this, I want to announce one more thing I have. Let me share my screen. We are trying to organize one in-person meeting. So it will be first international conference, Micro MBAT 1. So please block your date, 1st and 2nd June. Venue will be Delhi. And here we are going to micro dissection of embryology. So there will be so many things to learn. And with the national or international speaker will be there to interact. And it will be for limited people, we are not going to organize like 300, 400 people. It will be for limited, not more than 100 people. So please try to attend this meeting, and I invite all my moderator, faculty, and everyone here to join with us. Yes, thank you so much. Thank you, everyone. Thank you so much.
Thank you.
Thank you, sir.
Thank you, sir.
बढ़िया हो गया निशाद, तुम लोगों ने